Good morning, good afternoon, good evening, everybody, depending on where you are. Welcome to the technical sessions segment of the 2021 ISS Research and Development Conference. My name is Jim Way. I am Executive Director of the American Astronautical Society. It's great to have you here with us. We'll be joined later today by Alan DeLuna, the President of the Board of AAS and my co-MC for this event. For your information, the American Astronautical Society is a nonprofit technical society founded in 1954. Our mission is to advance all space science and exploration activities. We do that through our well-known symposia like the Goddard Symposium, the Glenn Symposium, the Von Braun Symposium. And that one is coming up October 12 through 14, so please check that out. We also have amazing technical conferences like our Astrodynamics Specialist Conference, our Spaceflight Mechanics meeting, and our Guidance, Navigation, and Control Conference. You can find out about all of our symposia and our technical conferences on our website at astronautical.org. We also partner with the folks at the ISS National Labs, or CASIS, on this conference, and AAS leads the effort on these technical sessions that we're starting now, and we'll be running over the next couple of days. So with that, make sure to connect with us on social media. Follow us on Instagram, Twitter, and Facebook. And I would like to thank our sponsors for the entire ISSR&D conference. First, our platinum sponsor, Boeing, who has been the lead sponsor of this event since it started back in 2012. We could not do it without you, Boeing, so thank you very much. And thank you to our silver sponsors, Airbus, KBR, Leidos, Northrop Grumman, Sierra Space, Space Tango, Teledyne Brown Engineering, and the Engineering and Innovative Technology Development Team at the University of Alabama. And also thanks to our exhibitors, Jacobs, Oceaneering, and TechShot. All right, so today there will be 6 sessions for the day today. There will be 15-minute breaks between each with a 45-minute break for lunch and an extended break at 12:30 p.m. Eastern. We will wrap up at 4:45 p.m. Eastern. During the event, we'll be referring to most times in Eastern, but we'll throw in a Greenwich Mean Time or Universal Time just to confuse everybody. You can ask questions using the Q&A tool located at the bottom of your Zoom screen. And please upvote the questions you want asked. You can do so by pressing the thumbs up button on any question that you see. And do also feel free to use the chat, engage with each other and have a fun time. So with that, let's jump right into our first session of the day. This is our first of 2 sessions on biology and medicine. We're lucky to have with us our session chair, John Love from the NASA Johnson Space Center. So John, if you could come on in and turn on your audio and your video. And your camera looks to be covered, John. There you go. That high-tech privacy tool, right? There you go.
That's it.
Well, good to see you, John. Good to have you here. And I'll turn it over to you to list off our 6 presenters or 6 presentations and presenters, and then we'll run the executive summaries back to back.
Okay, excellent. Thank you very much. So again, good morning, good afternoon, good evening to everyone. Thank you so much for joining us at this, the first of 2 biology and medicine technical sessions at the International Space Station Research and Development Conference. I will be introducing the names and presentation topics that we will be covering in this session. We have Carolina Marin de Evsikova from Bay Pines VA Healthcare System in Bay Pines, Florida, who will be talking about transcriptomics to detect changes in gene and transposon expression in flies exposed to microgravity and simulated hypergravity. We have Yupeng Chen from the University of Connecticut in Storrs, Connecticut, who will be covering DNA-inspired nanomatrices, enable tissue constructs and tissue chips for applications on Earth and in space. Chandran Sen from Indiana University in Indianapolis will be talking about an epigenetic trigger for induction of genomic plasticity following exposure to space environment. Another speaker and panelist is Gergo Mesohegi from the InnoStudio Inc. Company in Budapest, Hungary, and he will be covering effect of microgravity on COVID-19 drug formulation on ISS. Another presenter panelist is Giada Graziana Genci from the Istituto Italiano di Tecnologia in Pontedera, Pisa, Italy, talking about administration of antioxidant nanomaterials in simulated microgravity, the ESA IIT Intergravity project. And our sixth presenter panelist is Antonio Rampoldi from Emory University School of Medicine in Atlanta, Georgia. And he will be talking about generation of cardiomyocytes from human-induced pluripotent stem cell-derived cardiac progenitors expanded in space microgravity. So we have a very exciting agenda. We're looking forward to hear the presentations and then discuss the topics with the presenters. Again, please, if you have any questions, please enter them in the chat. With nothing else, welcome again, and thank you very much, Jim.
Okay, we're now going to run the executive summaries.
Thank you for attending my talk on transcriptomics and transposons. Adaptation of an individual organism to a new environment, such as spaceflight, is achieved by gene expression changes. Adaptation to stress induces a repertoire of transcription factors, as well as epigenetic changes to the genome of cells, such as DNA methylation, histone modifications. These changes consequently reactivate transposable elements residing in genomes. We analyzed RNA-seq data from flight and ground-based studies from NASA Gene Lab using a novel informatics pipeline, transcript and transposon signature analysis, also called T-TESA, to test our hypothesis that stress from adaptation to microgravity or hypergravity alters transposons and gene networks for biological processes in larval and adult flies. Our bioinformatics workflow processes experimental RNA-seq data through quality control, analysis of gene networks or transposons, and visualizes the results. Larval flies exposed to microgravity from spaceflight decrease gene networks regulating cuticle growth and development, energy production, oxidative reduction in mitochondrial pathways, and tyrosine metabolism, a building block of many peptides and proteins. Upregulated gene networks include morphogenesis, organ growth, chitin breakdown, and increase immune system and immune responses, as well as host defenses. These latter responses indicate that transposons may be involved. Numerous transposons significantly changed after microgravity compared to ground controls. Individual LTR retrotransposons are indicated in red, which can create new gene products in the genome. Larval flies exposed to microgravity from spaceflight decrease gene networks regulating cuticle growth and development, energy production, and tyrosine metabolism, a building block of many peptides and proteins. Some transposons were altered up to 250-fold from microgravity. Notably, the most differentially expressed transposons were long terminal repeat transposons, as shown in red and blue, which resemble retroviruses and can propagate readily within the genome because these transposons encode their own reverse transcriptase. Among transposon groups, long terminal repeat retrotransposons were the most prominent, with expression differences ranging from 8 to 100-fold between ground control and spaceflight. Fly larvae exposed to simulated hypergravity surprisingly also have downregulated gene networks for cellular respiration and mitochondrial activity and function, whereas gene networks for protein translation, stress, immune, and defense responses are upregulated. Hypergravity affected fewer transposons with more subtle changes in expression levels compared to microgravity studies. Notably, all 24 differentially expressed transposons were retrotransposons and almost exclusively the long-terminal repeat retrotransposons. Our novel transcriptomics t-tested pipeline detects meaningful gene and transposon expression changes driving biological processes arising from adaptation to spaceflight. Both microgravity and simulated hypergravity suppress gene networks for mitochondrial function while inducing networks for stress, immune, and defense responses. As transposons invoke damage control, their expression may drive some of the immune, antiviral, and other host defense mechanisms to protect genomes against threat. These studies support our previous work that spaceflight alters transposons and downregulates genetic networks for muscle activity, mitochondria, and growth in Japanese ricefish Medaka. These studies facilitate research to understand the role of retrotransposons in disease and serve as translational model for human exposures from space exploration and space tourism. I thank the American Astronautical Society for the invitation to present our studies.
Hello everyone, today I'm going to talk about our project developing DNA-inspired nanomatrix for cartilage tissue construct and tissue chips. The motivation of our study is to overcome the biomechanical loading problem on Earth and in space. On Earth, patients who suffer from movement limitations, they have abnormal biomechanical loading on cartilage, and such biomechanical loading can cause the degeneration of chondrocytes and cartilage. In space, the absence of biomechanical loading caused by microgravity can also damage chondrocytes and cartilage. Importantly, that the cartilage is the tissue cannot self-regenerate, so cartilage damage can be permanent. To overcome this biomechanical loading problem, we want to create a cartilage tissue construct and study therapeutic approach to overcome this problem. To create a cartilage tissue construct, we need to identify the biomaterial scaffolds. Conventional solid scaffolds that are not injectables, they have limitations for applications in difficult to reach locations, such as deep tissue injuries or the tissue chip microchannels. Hydrogels, on the other hand, they are injectable, but they are semi-solid material. They don't have the solid surface for cell anchorage, which could be a limitation in space. Here we develop an injectable nanomatrix. They can selectively promote the cell anchorage and functions for cartilage tissue engineering and cartilage tissue chip. So the injectable nanomatrix was developed by using DNA-inspired genus-based nanotubes that are designed and synthesized in our lab. And we will develop them as injectable material for tissue engineering and tissue chip. As shown here, this is an example of Matrix-3 nanomatrix. The nanotubes dissolved in water as a transparent solution injected into another transparent solution that has Matrix-3 molecules. And when we inject them together without using UV light or any chemical initiator, we can start to see a formation of those solid fibers in the solution. And those solid fibers are nanomatrix. And we can see those solid fibers are very flexible, so they can be injected into any difficult to reach locations. And more important that we discovered those nanomatrix, not only they are injectable, but they are very good at improve cell adhesion and anchorage. This is for the cartilage cells, and also significantly improve the cell proliferation compared with the GBNTs alone or the matrix alone or the negative control. And we also look at their long-term functions. We look at the aggregate expression, COL2 and SOX9. The nanomatrix group all showed excellent or significant improvement of those gene expression. And more important that by using this injectability of those nanomatrix, we can inject them into the microchannels of tissue chips. So here is the tissue chips we developed for cartilage tissues and using our nanomatrix. And the middle channel are the nanomatrix for cell anchorage functions. And we also look at the tissue chip functions. And again, this nanomatrix group has dramatically increased those cartilage-related gene expression compared with negative controls. So as a summary, that we have developed a solid and injectable nanomatrix to create a tissue engineered construct as well as can be used for cartilage tissue chips. As an acknowledgement, we would like to thank NSF and the ISS National Lab for funding. We would like to thank everyone who helped and support this project, especially my graduate student Ian Sanz and An Yao, who conducted this work. Thank you.
Good morning. I'm Chandan Sen from the Indiana Center for Regenerative Medicine and Engineering at the Indiana University School of Medicine. I'm going to today, on behalf of our team, as seen on the screen, share some of our findings on the ISS Rodent Mission 8. Now, mostly discussed with respect to prokaryotes, genomic plasticity, the notion, is also applicable to mammalians. And in particular, it has special significance in the context of adaptation, and this has been extensively studied. Now, as it relates to the molecular processes underlying genomic plasticity, one major foundation stone are epigenetic alterations. These happen fast, and therefore rapid adjustment to the environment is mediated through processes of epigenetic alteration. Here is a summary of the Rodent 8 mission and our study. There were 27 mice in total, of which 10 were maintained as ground control. 17 left the Earth, of which 10 were dissected in orbit and 7 returned back to Earth. In this case, the hind limb skin, a highly adaptive organ, was isolated and studied for their global 5-methylcytosine levels. Immunohistochemical analysis were done. Their methylome was analyzed, as well as mRNA-seq analysis was performed. What was noted in these cohort is that about a fourth of the animals that left the ground were responders. In other words, in these animals, we could see significant global demethylation, as you can see. So in black, you see the ground controls. In blue, you see the non-responders that left ground. But in red, you see those cases where you have substantial response to exposure to space environment. In these cases, methylation levels, so in this case, the abundance of 5-methylcytosine substantially decreased. Now, interestingly, this decrease was primarily noticed in the epithelial compartment, and also in the myeloid compartment. Myeloid cells are very well known for their plasticity, and as you see these types of demethylation, they are known to acquire substantial plasticity, as a result of which they could now have significant changes in their cell state. And interestingly, it once looked at the vascular endothelial compartment, no such changes were seen. So the demethylation changes that we are reporting happens in specific compartments of the tissue. Now, if one took a look at all the genes, one would see 56.89% of hypomethylated genes in the non-responders versus ground controls. But in the responders, these were 64.32% of the genes were hypomethylated. And if one carefully looked at every single gene, one would find 590 differentially methylated regions in the non-responders, whereas in the responders, you would see 1,278 differentially methylated regions in the responders. Now, if one took a look at the different parts of the genome, one would see that this demethylation is not specific to the region, meaning that the demethylation was comparable in the promoter, intron, as well as the exon sites. Now, these genes were then taken through ingenuity pathway analysis, and the top two pathways, hypermethylated pathways, were discovered to be NF-kappa B and HIF-1 alpha, both of which are known to be connected with several adaptive processes. In responders, because of the observed demethylation, two major pathways were favored. One was that of angiogenesis, and the other was that of VEGF signaling, as shown here. Finally, our results lead to a paradigm where we basically show that epigenetic gene silencing opposes tissue plasticity. So in fetal conditions, in fetal tissue, you have the least epigenetic gene silencing. And as you progress through life, in the complicated adults that are suffering from disease or stress, you have maximum epigenetic silencing. With that, you have the least tissue plasticity. So it seems that adult animals exposed to space environment is turning back in time and acquiring tissue plasticity by losing methylation of certain genes. So epigenetic gene silencing is dampened and plasticity is enhanced upon exposure to space environment. Thank you very much for your attention.
Good morning. This is Gergő Mezőhegyi speaking. I am the head of the Space Chemistry Division at our company in Studio, Hungary. And it's a great pleasure to be here at the ISS Research and Development Conference, even virtually. I'm going to now present our results and I think very interesting findings related to COVID-19 drug formulation on the International Space Station and the effect of microgravity on the drug formulation process. Drug discovery in space in general is becoming a very important and exciting issue in the field of space research. We think that both drug discovery and performing chemistry in space can offer a great opportunity for both the benefit of human mankind on Earth, but also for space travelers in the close future. Microgravity may ensure faster or even more efficient reaction routes, considering active pharmaceutical ingredients development. Also, it's possible that novel reaction products or chemical compounds can be developed under microgravity conditions, which are either impossible or difficult to synthesize under Earth conditions. The active pharmaceutical ingredients production in space is becoming soon a very important issue to be solved, since astronauts cannot take all the necessary medications with themselves for longer space trips, considering the actual stability problems occurring. In this study, we try to combine the microgravity environment with advanced drug research related to the actual world health problem of COVID-19. We selected remdesivir as target of our experiment, which is the active compound of one of the most important and most widely used drug nowadays against COVID-19. This is marketed as Veklury in the United States in form of a cyclodextrin complex. Unfortunately, this medication has limiting factors, considering that the cyclodextrin can accumulate in the kidney, so certain groups of patients cannot take this medication at the moment. It's evident then that improvement of the active compound cyclodextrin ratio could allow in increasing the treatable client group. Unfortunately, this could not yet been achieved under Earth conditions, and microgravity offers a possibility to reach this improvement. So we prepared our samples in a frame of an international collaboration where our Japanese partner, Japan Manned Space Systems Corporation, took our samples within their system so-called Kirara to the European Space Agency's Columbus module, where the samples were placed into the facility of ice cubes supported by Space Applications services. Samples were taken by the SpaceX Dragon to the International Space Station and spent a bit more than 1 month in the space station. For summarizing the results, an unexpected and important result of the study was that in one of the 2 space samples, significantly less degradation rate of remdesivir in the complex was observed. This suggests that the complex formation, perhaps also the remdesivir degradation, took place via different chemical mechanism in space and on Earth, which, besides potential clinical significance, may induce a very new approach to chemical and drug discovery studies in space, including the exploration of diverse decomposition routes of molecules in space and on Earth, and also stabilization studies for active pharmaceutical ingredients. Thank you so much for your attention. Please visit our full briefing for more details of our study. Thank you.
This executive summary presents an overview of the talk entitled Administration of Antioxidant Nanomaterials in Simulated Microgravity, the ESA IIT Intergravity project, concerning work performed by the Smart Biointerfaces Group of the Italian Institute of Technology with the support of the European Space Agency. The project aims at testing innovative nanotechnological antioxidants against muscle mass and force loss, typically affecting astronauts in space due to mechanical unloading and exposure to cosmic radiations, as well as individuals on Earth due to aging and pathology onset. For their self-renewable and biomimetic antioxidant effect, serum oxide nanoparticles were synthesized by wet chemistry process, the main steps of which are depicted in this slide, and were characterized before and after coating with fetal bovine serum, necessary for stabilization in aqueous medium. This class of inorganic nanoparticles is indeed well known for its superoxide dismutase and catalase-like activity, which may prove very useful to long-lasting alleviation of oxidative stress resulting from overproduction of pro-oxidant chemical species within biological systems. Raman spectroscopy and X-ray diffraction confirmed the obtainment of nanoparticles with the desired chemistry, as well as their nanometric size. The latter was confirmed by transmission electron microscopy, revealing that nanoparticle size was indeed below 10 nanometers. Thermogravimetric analysis demonstrated that nanoparticles underwent a significant adsorption of serum, thus providing suitable stabilization in aqueous medium. Nanoparticle dispersion stabilization induced by serum was also shown with dynamic light scattering and zeta potential analysis conducted on different aqueous solutions over a period of 6 days. Photometry revealed that nanoparticles exhibited an excellent antioxidant property, quantified in Trollocs equivalents with value much higher than those of organic soluble antioxidants. The latter are known to have a short half-life in biological environments and therefore need repeated administrations, often incompatible with long-term spaceflight. Biological experiments conducted with human skeletal myoblasts demonstrated that the cultures tolerated a wide range of nanoparticle concentrations, undergoing regular proliferation within a period of 4 days under normal gravity. The cultures were also exposed to simulated microgravity conditions on a random positioning machine. Flow cytometry analysis upon staining of cells with a probe that becomes fluorescent upon interaction with certain pro-oxidant chemical species revealed that nanoparticles mediated alleviation of a mild oxidative stress associated to random spin rotation within 24 hours. Preliminary investigations concerning interaction of serum oxide nanoparticles with cells under simulated microgravity were conducted by culture of mouse myoblasts with commercial nanoparticles and by performing immunostaining and confocal microscopy. Image analysis by quantification of Pearson's correlation coefficient between nanoparticles and proteins involved in endocytosis revealed that nanoparticles are actually internalized under simulated microgravity, locating in the cytosol close to the nuclei, but with very modest recruitment of caveolin and clathrin. Nanoparticle internalization mechanisms and oxidative stress alleviation during myoblast culture, including differentiation under simulated microgravity, will be object of thorough investigations with a human model in the next future. To conclude this, I would like to acknowledge the support of ESA, of my co-author, and of many colleagues at IIT. Thank you all for your attention.
This investigation was built on a previous simulated microgravity experiment that we conducted on Calibration progenitor cells that were maintained for 72 hours in a random positioning machine, which simulated through random movement wetness. Cardiosphere had a better diffusion of nutrients, low surface tension, turbulence, and shear stress. We observed that this simulated microgravity promotes survival and proliferation of cardiac progenitor and also promotes cardiomonocyte differentiation. Our objective was to examine how spaceflight affects the expansion and differentiation of cardiac progenitor cell, specifically examining the proliferation of cardiac progenitor, analyzing the molecular profile of cardiac progenitor, and characterize cardiomonocyte derived from cardiac progenitor after exposure to microgravity. In order to do this experiment, we cryopreserve cardiosphere and inject them into culture bag inside this MVP multi-use variable gravity platform from TechShot. This MVP facility contains two carousel, each one holding a 6 cell culture module, and it can operate at microgravity and 1G through centrifugation. Medium change was automatic through pumps for 22 days. Microgravity cardiosphere were on average 4 times bigger compared to the 1G cardiosphere. In the microgravity cardiosphere, few cells were positive for the dead cell dye etidium, while the majority of 1G cardiosphere were fully positive for etidium. Nucleic count on a live cell show a 20 times difference between the microgravity and 1G sample. Cells were analyzed through RScan for cell purity and cell proliferation. Cell purity was assessed with cadmium marker NKX 2.5. A high percentage, more than 90% of NKX positive cell was observed in both conditions. Cell proliferation was assessed with proliferation marker Ki67. Around 7% of microgravity cells were positive for Ki67, but no Ki67 positive cells were observed in the 1G cell. Later, real-time PCR showed that several genes related to cell proliferation were significantly upregulated in the microgravity cell. Microgravity cardiac cell had a reduced abnormal calcium signaling compared to the 1G cell. Also, microgravity cardiac cell had an increased peak amplitude and an increased ramp speed. This indicates that space microgravity improves contractility and calcium signaling. Aliquot of cardiac sphere were collected 3 days after being towed aboard the International Space Station and upon return to Earth in order to observe the short and long-term effect of microgravity on cell. In the short-term sample, 195 genes were significantly upregulated and 207 were downregulated in the microgravity sample. The gene expression analysis of the long-term effect of microgravity on cell is currently still ongoing. In the microgravity cell sample, most of the upregulated genes were involved in cell proliferation, cell regeneration, air development, and cadmocyte contractility. The downregulated genes instead were mostly involved in mediating stress response, inflammation, apoptosis, or in pathway that lead to differentiation in the other cell type instead of cadmocyte. Astronauts successfully towed and cultured cardiac progenitor sphere on the International Space Station. The MVP system was able to simulate Earth gravity, allowing it to determine the effect of space microgravity on cardiac cell. Viable beating cadmocytes were able to safely return to Earth for further study, and we determined that space microgravity positively affected cadmocyte differentiation, proliferation, cell survival, and cardiac function. The RNA-seq analysis on a cell under short-term exposure to space microgravity show gene and pathway involved in cardiac differentiation and proliferation. I would like to acknowledge our collaborators, the people of my laboratory, Emory University, Georgia Tech, TechShot, and Cases.
All right, so that does it for our executive presentations for this first session. So John, if you want to come back in again, and we'll get going with the discussion and Q&A. And all of our presenters, please go ahead and turn on your video, but keep yourself muted unless you're talking. Yupeng, good to see you. Antonio, Giorgio, Chandan, Giada, great. Looks like we have almost everybody. We might be missing one, but John, take it away, please.
Okay, excellent. Hello, everyone. Great to have you. Excellent presentations. Thank you so much for participating. So we're going to start with questions that were submitted through the question and answer feature. And some of those questions appear to be generic. So I'll open the questions to the panelists to see if any or several of you or all of you would like to take a stab at answering them. So let's start with the first of the questions. It is a question that asks about the effect of microgravity or space on human DNA. It's a generic question, so if any of you would like to take it, we'll hear what you have to say. And again, multiple panelists can respond to the same question. Effect of microgravity or space on human DNA. I know some of your presentations hint at potential countermeasures, extrapolations from model organism studies, as well as data that is based on research studies that has led to what you are proposing. So if you want to link it to your particular presentation or study as part of the rationale for your research, please go ahead and do so.
Yeah, so maybe I will jump in first. I'm a bioengineer, not a biologist, but I do know that at a microgravity environment, usually it promotes cell proliferation, and this is through several mechanisms. And one potential is also improve the DNA duplication. And another thing is in the space. Although on the International Space Station, we have already protected the astronauts very well, but still there a possibility of getting at the space environment. There are potential to cause some damage to DNA and the cells, but usually on the International Space Station, this type of impact is minimized. But this is my what I learned from about the influence on DNAs.
Mm-hmm. Excellent. Thank you very much, Dr. Chen. Anyone else wants to make any comments? Jim, I think one of our speakers was listed as apparently as a participant and not a speaker. Yep. So I don't know if we've been able to join her.
She's in there now. She's in there now.
Okay,
Carolina
excellent.
is in. So great.
So maybe we can extrapolate that generic question. We have another question that is also related to that. How does space environment affect different genes? So let's again take those questions and see if the panelists would like to answer those questions in relation to their own studies and their presentations. For instance, we can start with Dr. Evsikova. If you'd like to comment on transposable genetic elements and how they relate potentially to the stressors of the space environment or microgravity, and again, from the findings of your analysis, how you can expect that such an environment impacts, again, the characteristics of transposable elements.
Yes, what we found is that there seems to be an upregulation in a specific class of transposons known as long terminal repeat retrotransposons, which can actually essentially rewrite the genome. These types of transposons are known to act as alternative promoters. They can drive neighboring gene expression, and they can make new transcripts. And so what we have seen is that there was potentially new gene networks that were affected. And it's possible because of how transposons evoke stress and immune responses, those could be involved. And we saw such an upregulation of many of the LTR retrotransposons, as well as they were anywhere between 8 to 100-fold increase. So we do think there is a genetic, some of the genetic remodeling of networks may be driven by the transposons. And we've now seen this in 2 different species of animals that have been exposed to the living in microgravity.
Mm-hmm. Excellent. Thank you very much. Dr. Sen, did you want to?
Yeah, I'd like to add there
expand on that question.
That excellent response. I'd like to add that based on our findings, we see a clear individual variation, what we are calling responders and non-responders. Take it the transposons, or take it the epigenetic pillar of genomic plasticity. Clearly, all of these are responding. And in our case, when we are specifically focusing on methylation status, it is clear that you have responders and non-responders, and the difference is striking. In other words, the non-responders are very much akin to the ground controls, whereas the responders are strikingly separate, not 10-20%, but strikingly separate. And this allows for more adaptive processes to play in. Now, what exactly is the outcome of that functionally remains to be seen, but clearly, with nothing done and everything done the same to all these animals, we do see a fourth of the animal falling in that responder category.
Excellent. Thank you so much. Anyone else wants to comment on differential expression or of genes or general impact of the space environment on genomics? Go ahead, Dr. Rampoldi. Thank you.
Yeah. We saw, first of all, our experiment was made in a way that we wanted to see what was the real effect of microgravity. So we had control not on Earth like normally is done, but we had this cell in a centrifuge on the International Space Station, so that it was simulating the Earth gravity, and we were able to see that there was upregulation and the regulation of several genes in clearly different pathway. We, as you can see, as you saw in my presentation, gene associated with cell proliferation, gene associated with cardiomyocyte development were mostly upregulated in our microgravity sample, and we also saw downregulation in gene associated with apoptosis, inflammation, or cellular stress. And we also saw that in the 1G sample, a few genes associated with ionizing radiation were also upregulated. So it's microgravity, it's really changing the pathway and gene in a clearly significant way. And it's not only the microgravity in space that is making this alteration, there is also this space radiation and other background event that can apply some clearly big change in the cell.
Excellent. Thank you so much, Dr. Rampoldi. Any additional comments? I wanted to see maybe Dr. Mezuhegi or Dr. Genchi. Would you have any comments on differential gene expression? I'll increase the scope of the question based on another question we received, and that is like, how do you keep your genes normal? So let's expand this at other things that you're working on in general, and that's the development of potential countermeasure applications or research that can translate into such kind of approaches that would minimize or completely eliminate the impact of the environment on genetic changes. Anyone would like to talk about that? Or we can start with Dr. Mezuhegi or Dr. Genchi. Any comments from your end?
Yeah, thank you. This is Gergő Mezuhegi here. We would be much better prefer drug discovery related questions, if any, instead of the biological gene expressions. Thank you.
Thank you very much. I think Dr. Zen had raised his hand.
Yeah, I'd like to just make a comment here that whatever we know about terrestrial biology, including normal DNA that we understand under Earth conditions, should not be placed as a burden as to what is normal, because we are talking about a new natural environment and we are a natural product. So that interaction may have its new set of normals. So any deviation from terrestrial conditions should not be viewed as abnormal, could also be viewed as some level of adaptive process. So it's a mindset issue as to whatever departs from terrestrial condition is not necessarily injury, pathology, and abnormal. And so I think it's important to put observations in that context.
Very good point. Thank you very much. And that's something, again, some of your studies have to do with adaptation and plasticity. And that is a good point, that that may be the adjusted normal in quotations when there is a change in that new environment. I think Dr. Chen wanted to comment. Go ahead.
Yes, thank you very much. Excellent discussion. So I want to add some comments from our own experience that, as I mentioned, that the DNA could be altered that at microgravity, at a combination between microgravity as well as space radiation. And what we can do is I already identified the cell signaling change and the microgravity environment. For example, that those cell signaling can increase inflammation, as I've already mentioned, and decrease DNA repair. And that may also damage, well, not damage, but also, as Dr. San mentioned, that alter the cell morphology. What we do is we have using nanomaterials to help the encapsulation and the anchorage of the cells to, for example, we can improve the cell integrin signaling so that it can stimulate better DNA repair, reduce apoptosis, as well as also give a cell some signaling. We also use microRNA try to transfect into the cells to give the cell a signaling that similar when they have in the normal gravity environment, so that we want to use this type of chemistry or biochemistry method to try to overcome the impact of microgravity. But this is an ongoing discovery, and this is what we are working on. Thanks.
Excellent. Thank you very much for your comments, Dr. Chen. Very exciting. Looking forward to your findings. Thank you so much. Let's see, Dr. Genchi,
yes.
Go ahead.
Yes, I would like to add just a few observations concerning the effects of microgravity on DNA or on the overall nuclear content. Based on our experience with serum oxide nanoparticles, the commercial ones that we sent on board the International Space Station with a past project from our group, we know that these nanoparticles can help protecting the nuclei, and this was based on the observation of the regulation of transcriptions of some genes involved in nuclear integrity, like lamin and histone, a protein that's involved into let's say packing of the DNA. Because as far as we know, DNA can be damaged. It can also be transcriptionally regulated by the microgravity, because it affects the packing actually of DNA. So it's very important protecting this compartment and from the noxious effects of both microgravity, but also of cosmic radiations, because radiations can have direct effects on the DNA by producing damage to the DNA strands, which can be partially repaired by our repair system. That's why we would like to help, we would like to support this activity. But they can also produce noxious effects, radiations I mean, by producing an excess of oxidative species, also called free radicals. And that's why we would like to administer antioxidants so as to have overall protective effects on biological functions. The advantage of having some inorganic nanomaterials like serum oxide nanoparticles is mainly provided by their long-term effect, which can support long-lasting activities in space. This was the aim of our study, and we would like to support human activities in a long term, especially during longer space flights like interplanetary travels.
Excellent. Thank you very much for your comments. And again, like with many of these studies, applications can also go to challenges that happen here on Earth. in your case, addressing oxidative stress that may be involved with the development of atherosclerosis or other things like that. Your approaches may potentially have applications for other maladies that happen here. And it's exciting to hear that your approach is self-regenerative, which is very interesting. There is another question that came up for one of the speakers, and I will ask to all of you. Actually, Dr. Sen had his hand raised. Apologies. Go ahead.
Yeah, no, thank you. And I just, that excellent answer, I'd like to qualify that with an observation. And the observation is in terrestrial biology, we are quite familiar with conditions where injury is followed by adaptation. For example, if we take physical exercise that we pretty much all are engaged in to keep fit and healthy, in physical exercise, we have copious amounts of oxidative stress leading to oxidative DNA damage, and those oxidative DNA damage products passing out to the urine. Pretty much every competitive athlete, that has been a part of what we have studied otherwise. So in other words, the challenge here would be to allow enough injury to enable adaptation, but then to limit injury to a point beyond when it is pathogenic. So that qualification, I think, would guide much of what we are seeking to do with respect to therapeutic intervention.
Great comments. Thank you very much again, Dr. Sen. So I will ask perhaps one final question since we have 3 minutes left. It came for one of the speakers, but I'll ask all of you. And I guess that regards how some of these changes may persist or not when potential spacefarers return to Earth. If there are changes that occur in the microgravity environment, do they persist once they are away from the weightlessness of orbital spaceflight? Are they reversible? What are your thoughts about that? And if they need to be addressed here on Earth, or do they just disappear again as another adaptation process may be occurring? So I'll open the floor to discuss that aspect of return.
So I can, because I'm working on cartilage, I know that the cartilage tissue itself has very limited self-repairing or regenerating ability. So if there are damage and the microgravity environment that cause a cartilage defect or degeneration, it will be very difficult to repair completely. Although there will be some repair, but the newly repaired tissue is at least as a natural condition is never match the original cartilage tissue. So this is at the tissue level that there are the changes.
And a quick comment there would be that whatever we think, cannot happen with the human body or, say mammalian life, are all limits that we know under terrestrial conditions. We need to find the new normal of space conditions as to are those limits. In many cases, physiology and genetics plays out differently in space. And we need to understand that to be able to extrapolate limitations of terrestrial condition all the way to space. So that context, I think, is very important to keep in mind as we talk about whatever we know is in Earth actually applies in space, cannot be just an assumption.
Excellent point. Thank you very much. We have about a minute left. Any additional comments from anyone? Again, about potential changes or adjustments of impacts of the space environment on our cells and genomics once astronauts return to Earth. Okay, I think we actually reached the end of the session. It was a set of fantastic presentations. Thank you so much to the speakers for the outstanding jobs and excellent discussion. We really appreciate your participation. Back to you, Jim.
Excellent, thank you. Oh, sorry, Carolina, did you want to get in one last comment there?
Yes, for the question of adaptation to gravity here on Earth, what our preliminary analysis indicated that while Drosophila seemed to readapt in many of the gene networks, we found that Medaka, Japanese rice fish, don't seem to readapt within at least a short period of time. We don't have much data looking at readaptation like a month later. We only usually have 24 to 72 hours. And we look for that. So we were surprised actually at the flies how quickly they were readapting, whereas with the Medaka, they were not. So it's hard to know. I wish there was just more data available.
Yeah, how interesting. That may be the basis for future studies, but very exciting findings and wonderful prospects. Thanks again for all your comments. Any additional comments from anyone? Thank you very much again. Back to you, Jim.
Excellent. Thank you, John. Thank you to all of our panelists so much. That was very interesting. Great stuff. Great way to kick off the day. So we thank you all very, very much. It was great. We'll now take a break until 10:15 Eastern, when we'll reconvene back here for our second session on biology and medicine. Alan DeLuna, the president of AAS, will be your host for that session. So we'll see you in about 13 minutes. Thanks, everybody.
Thank you. Bye-bye.
And I am the CSO of ACOR.
John, this is Alan DeLuna, and I know the world is listening, but I wanted to tell you that we've got Ali Hong and Cynthia Brazel that have not yet logged in. If they log in, we will bring them in as quickly as we can, and we'll start our session in 2 minutes.
Sounds good, Alan. Thank you so much. Copy.
Good morning, everyone. We're now time for our second session of the day. That's Biology and Medicine session number 2. Our chair for today is John Love of NASA JSC. He's deeply ingrained in the ISSR&D program. John, why don't you come on and tell us who's presenting to us today?
Okay. Can you see me?
Yes, sir.
Okay. Good morning, good afternoon, good evening to everyone, and thank you so much for joining the second Biology and Medicine session at the ISS Research and Development Conference. We're very excited about all of these presentations, and we are very grateful to the participants for coming in and staying also for a panel session. So today we have the following presentations. Daniel Moland from King's College London in Arundel, West Sussex, United Kingdom, will be talking about development of the Flexidid approach to investigate antimicrobials aboard the International Space Station. Then we have Ali Huang from Mini PCR in Cambridge, Massachusetts, speaking about Biobits, cell-free protein synthesis to support on-demand protein production in spacecraft. Following that presentation, Cynthia Brazel from ECOR Inc. in San Diego, California, will be talking about ECOR's antimicrobial biofilm inhibitors for environmental control and life support. That presentation will be followed by a talk by Rosa Santomartino from the University of Edinburgh Center for Astrobiology in Edinburgh, the United Kingdom, covering biorock and bioasteroid. What did we learn on microbe-mineral interaction in space? Then we will have Jeffrey Ebersole from the University of Nevada, Las Vegas School of Dental Medicine. He will be speaking about oral biofilms in space, a novel microfluidics device for biological studies on the International Space Station. And our final presentation for this session will be by Eric Yost from Grand Canyon University and Stellar Quest Institute in San Diego, California. And he will be covering the photochemical and electrical effects on a microbial fuel cell in microgravity. So we have 6 very exciting talks, and we're ready to begin. Again, thank you so much to the panelists for staying for a question and answer session as well as discussion, and to all of the other participants and attendees. If you have questions, please go ahead and submit them through the question and answer feature. That's all I have. Going back to you, Alan.
All right, thank you, John. And for Daniel, I feel an affection for Daniel. My son graduated from University of Brighton. I think that's in East Sussex instead of West Sussex. But you know, you have to feel good for the people coming in from the UK. So now we're going to start, and we're going to play all of the 5-minute executive summaries. And when they are done, we're going to jump in, and John's going to lead the Q&A. And John, we do have all 6 panelists on now. So Abigail, if you'll run our presentations, please.
Hi everyone, my name is Daniel Molland and I'm a visiting researcher at King's College London and a lead scientist at the International Space School Educational Trust. Today I'm talking about the development of the Flexidid approach to investigate antimicrobial activity in microgravity. This will cover the development of a new approach that we have developed at King's College London that enables the mixing of antimicrobial and bacterial species of interest within microgravity that complies with NASA safety containment regulations. When developing experiments for the International Space Station, there are a number of constraints that first-time users may not be aware of. These include a limit on number of repeats, a problem with fluid management, and most importantly to antimicrobial studies, the constraints relevant to layers of containment, which prevent direct contact between astronauts and biological studies. The ISET Payload Research and Development Team came across this problem very early on when we found experiments conducted using terrestrial methods did not work in microgravity. This was because when these experiments were set up at the Kennedy Space Center and then launched, the agents of interest would diffuse across the entire agar growth media, preventing any observations of dose-dependent antimicrobial activity. To address this, we have developed a new method that allows the mixing of antimicrobial species and species of interest to be mixed only when they are in a microgravity and which is resistant to the stresses of launch. This thus allows the investigation of antimicrobial activity in microgravity only when initiated through astronaut contact. The Flexidid approach is a very simple technique that allows the mixing of agents of interest loaded onto absorbent disks to be mixed with species of interest aboard the International Space Station that complies with the relevant containment regulations. On the top section shown here, we see a simple layout of this with a 5.9 cm diameter Petri plate poured with a growth media and 2% agar. This is then spread with an agent of interest. The top side of the Petri plate, which is normally used in studies, is discarded and replaced with sterile parafilm, which is loaded with up to 5 absorbent discs, with each disc containing an agent of interest. These are bonded to the parafilm using a volume of 2% agar. The volume can be adjusted based on how much fluid is placed in the absorbent discs, with more fluid in the discs, meaning there needs to be more agar to ensure the disc stays fixed to the parafilm during the launch process. The sterile parafilm loaded with the discs is then placed over the agar growth media and sealed with a further stretch of parafilm, creating a gap between the growth media with the bacteria and the parafilm discs. The discs can then be deployed onto the growth media when the experiment reaches the ISS by simply pressing down on each absorbent disc, bringing it into contact with the growth media when the astronaut activates it. The benefit of this system is that it can be done through containment layers very simply just by pressing down on the parafilm, thus ensuring that the agents can be mixed without introducing an expensive level of automation or damaging the containment layers, which are critical to ensure astronaut safety. Shown here is how this looks as a finished result. On the left, we can see how a pre-activation example with the discs still attached to the parafilm lid using the agar fixative. On the right, we can see this post-activation with this image taken by astronaut Drew Morgan, who had just pressed down the absorbent discs onto the reverse side of this plate, thus bringing the antimicrobial, in this picture adipic acid, into contact with the SRAS. This thus enables the study of the antimicrobial effect of this agent in a safe yet extremely reliable setup that has now been demonstrated across several launches. To finish off, I would like to acknowledge the collaborators on this work, particularly Dr. Julie Keeble, the ISET Chief Scientist, and Zoe Gaffin, the ISET Chief Payload Developer. As well as this, I'd also like to acknowledge the funders of our research, Ross Barber and Chris Barber. And finally, King's College London and Nanoracks, who are our hosting organisation and who we work with to launch our current experimental payload to the ISS.
Hi, I'm Dr. Ali Juan, and I'm with Mini PCR Bio. And today, my talk will cover cell-free protein synthesis technology and how it could be used on the ISS and other spacecraft. My full talk will go over what proteins are, followed by introducing cell-free protein synthesis, describe some really interesting applications of this technology here on Earth and how that could be applied to space, and finally give you a sneak peek of our plans to launch next year. I will briefly touch upon these points now in this summary. There are two big challenges in living on the ISS that biology plays a huge role in. The first is dealing with the impact of microgravity and its significant impact on biology and how our bodies work, all the way down to a molecular and cellular level. The second challenge is the limited resources, many of which are biological, so we need to also better understand how microgravity impacts how we might self-sustainably make these resources in space. Proteins are an essential part of all biology and can be really useful tools in a lot of different settings. Many therapeutics, like insulin for diabetes patients, are proteins-based. Proteins are also a very useful tool in biology research and experiments. And there are certain types of proteins that can help perform biochemical reactions that result in biofuels. In order to be more self-sufficient on the ISS or on longer-range missions where resupplies from Earth are not feasible, it would be useful to be able to produce proteins for all of these applications and more. Scientists here on Earth can make proteins for all types of applications. As long as they have the DNA that has the instructions for making a given protein, they can use that to make all sorts of proteins in a lab setting by using live cell cultures as their protein-making factories. But it's really hard to do this outside of a lab without a ton of resources, especially if you are in space on the ISS without the room, resources, or expertise to be able to carry out these cell cultures efficiently and safely. There is an alternative to using live cells as your protein-making factory, cell-free protein synthesis. I will go over how this works in more detail in my full talk, but briefly, all of the molecular machinery and materials needed to build a protein has been extracted and freeze-dried into a portable reaction in a small tube. Since there are no living cells in this tube, you need far less resources, equipment, and biohazard regulations to make your protein. All you have to do is add the DNA that encodes the protein you want, and then the reaction will make it. This system allows one to make proteins on demand at the point of care, such as making the proteins found in vaccines right at the site where they are administered and reducing shipping and storage concerns. Not only would these be useful on Earth, but also on the ISS as well, where shipping and storing medicines and therapeutics can be tricky. We can also leverage the cell-free system and use the protein it makes as a visual signal or indicator in diagnostics to screen for pathogens or other biochemical molecules, such as screening for contaminants in water. You can imagine a system like this water quality testing assay to be very useful in space, as water is a limited resource on the ISS, and being able to easily monitor its quality or the ISS environment in general for contaminants and pollutants would be important. So far, to my knowledge, no cell-free protein synthesis systems have been brought or used in space, but starting next year, that will change. Through the Genes in Space competition that is sponsored by Mini PCR Bio and Boeing, we will be launching our own cell-free system to the ISS, a system that I worked on personally called Biobits. I developed Biobits in grad school specifically for students to engage in hands-on activities to learn about DNA, proteins, and other molecular biology basics. And because it is a portable system, students and teachers were still able to do all of these activities remotely together in the past year or so. Furthermore, our students who entered our Genes in Space competition were able to use the Biobits cell-free protein synthesis system this year to propose biology experiments to be done on the ISS. To conclude our presentation, our winner this year, Celine Coquillard, will also discuss her project on Biobits as a cell-free assay to test water quality on the ISS, which will launch next year.
My name is Cynthia Brazell, and I am the CSO of ACOR. My background is in marine and medical microbiology, and my expertise is with contamination control in biofilms. Bacteria have hitchhiked up to the ISS, where they quickly develop resistance, degrade equipment, and contaminate life support systems, threatening astronaut health, long-term space travel, future space living, and cross-contamination back to Earth. The bacteria in the ISS water systems have become resistant to silver, the antimicrobial of choice, and form biofilm slime. And it has already resulted in loss of hardware, impact to system operations, and increased resupply. These images are of the water recovery system that recycles wastewater, including urine, back into potable water, and the image on the left is a pipe clogged with biofilm. To tell you why ACOR is uniquely positioned in this space, I will take you back to our origin story. I discovered novel bacteria from the coral reef environment that produce natural antimicrobials. ACOR's treatments either kill or inhibit the growth of a wide range of bacteria and fungi that contaminate surfaces and water, including several resistant strains. ACOR's portfolio also contains chemical analogs that mimic the natural chemical, and several are GRAS or FDA food additives. We wanted to understand if ACOR's treatments would be effective against 3 difficult-to-treat biofilm-forming bacteria isolated from the ISS water system, including the silver-resistant strain. The testing took place in 2 different phases, both at ACOR's lab and at the Marshall Space Flight Center. Phase 1 was a laboratory series of antimicrobial and antibiofilm tests. 8 of ACOR's treatments were tested to select the treatment that performed the best, and treatment A1026 was selected for Phase 2. For Phase 2, 5 55-liter tanks were filled with wastewater ersatz and the mixed bacterial inoculum. The schematic on the right is of the water processor assembly. The tanks periodically cycled back and forth from a transfer tank, mimicking the cycling process on the ISS. For Phase 2, ACOR's A1026 treatment killed 99.999% of the bacteria the first day and maintained activity for 15 months. Tanks 2 and 3 have the same concentration, and Tank 4 had a lower concentration. We also saw a dose-dependent curve in Phase 1 on the smaller scale studies. For biofilm prevention imaging analysis, we stained the live cells green with cyto9 green fluorescent nucleic acid stain and dead cells red with propydium iodide. The image on the left is the control, showing thick biofilm that stained green. The image in the middle is a dose of A1026 that inhibited biofilm formation but did not kill all the cells, and these cells are washed off easily. The image on the right shows a dose of A1026 that killed the cells, and you can see they appear red. To determine stability of A1026 in the tanks, a calibration curve of different concentrations of A1026 was developed, and samples were measured throughout the study. The calibration curve was used to determine the concentration of A1026 remaining in tanks 2 to 4. You can see the slight degradation of A1026 over 15 months. The amounts of the treatment that remained are more than sufficient to maintain antimicrobial and antibiofilm activity. ACOR won the NASA iTech Award in the Life Support category, and we are participating in the Catalyst program with aerospace prime contractors. The image on the right is a paper presented by Marshall at the 2019 International Conference on Engineering Sciences. This slide is of astronauts drinking recycled water on the space station. And NASA outlines in the Human Adaptation to Spaceflight, the role of food and nutrition, that the ability to reclaim water from urine will be an absolute requirement for exploration class missions. The impacts that this has are de-risking the threats to astronaut health associated with contamination, improving the integrity of equipment by preventing degradation, providing an option for a multifaceted approach, and overall de-risking the mission. I wanted to acknowledge the folks at Marshall and microbiologists at Boeing who provided the bacteria, and I wanted to thank the AAS, ISS, and NASA for this opportunity. Please contact me if you have any interest. Thank you.
And this is Alan DeLuna, your co-host for the sessions today. I wanted to jump in and make an announcement to make sure folks understand that they can go ahead and start writing questions for these presenters and authors. Just go down to the bottom of your screen, click in the Q&A bullet down there, and put together your question. Please make it a question and not a speech. Hopefully you type like I, and you type pretty slowly, so it makes my ability to make speeches on the Q&A tool very difficult. But we wanted to go forward here and continue with our presentations.
Good afternoon. My name is Rosa Santomartino, and I am a researcher at the University of Edinburgh. In this talk, I will show you the results from the ESA BioRock and the BioAsteroid space experiments performed on the International Space Station in 2019 and 2020. The data obtained show the potential roles of micromineral interaction and biomining for human space exploration. As the goal for space exploration expands toward establishing a growing presence in space, microorganisms could provide an invaluable support. In this context, biomining, which is the use of microorganisms to extract useful elements from rock, ore, and mine waste, could offer a way to obtain needed materials directly on a planetary body, rather than relying on a continuous resupply from Earth. To understand if we can perform biomining in space, we first need to test how space conditions present on planetary bodies of interest, especially gravity, influence microbial interaction with rock and mineral material. It is with this idea in mind that we performed the first two biomining experiments on a space station, BioRock and BioAsteroid, launched on the ISS in 2019 and 2020 respectively. For the first one, BioRock, we selected 3 heterotrophic bacterial species, Sphingomonas dissecabilis, Bacillus subtilis, and Cupriavidus metallidurans. The rock substrate selected for the experiment was basalt, cut into small slides, each one representing a separate sample. 3 identical sets of samples were launched on board the ISS and subjected to microgravity, simulated Martian gravity, or simulated terrestrial gravity. A 4th set remained on ground and was subjected to real terrestrial gravity. We first analyzed the final cell concentration reached in the liquid cultures. The results show no difference between the space samples after 21 days of growth. This suggests that biotechnological application on Mars, Moon, or other celestial bodies will not be negatively influenced by the gravity condition. From the biomining perspective, we focused on 2 types of elements: rare earth elements, which are a group of metals with an enormous impact on our everyday life as they are present in any piece of modern technology, and vanadium, a metal widely used in structural materials and in alloys. Analyzing the samples from Biorock by mass spectrometry, we discovered that one of the three organisms, Sphingomonas dissecabilis, was able to effectively biomine rare earth elements from basalt in all the gravity conditions tested. While two microorganisms, Sphingomonas dissecabilis and Bacillus subtilis, were able to biomine vanadium in all the gravity conditions tested in space. These important results demonstrate that biomining is, in principle, achievable under a range of gravity conditions. After the success of Biorock, our group in Edinburgh recently completed a second space biomining experiment called Bioasteroid. We investigated how a bacterium, a fungus, and a mix of the two interacted with real meteorite material. In this case, a set of samples, including non-biology control, has flown on the ISS and was subjected to microgravity, while the ground control set was subjected to real terrestrial gravity on Earth. Analyses are still ongoing, but we already have some exciting results. Meteorite rock pieces from the space samples were analyzed by scanning electron microscopy. Bacterial samples presented interesting rod-shaped features, here indicated with red arrows, that may indicate effective bioinformation on the meteorite rocks in microgravity. However, some non-biological control presented similar features, and therefore further analyses are required. Fungal-containing samples showed an effective formation of mycelium on the meteorite rocks in microgravity, here indicated with blue arrows. In conclusion, with Biorock and Bioasteroid, we provided the first demonstration of biomining on a space station, under a range of gravity regimens, using organotrophic bacteria and fungi. We demonstrated that final cell numbers are not negatively affected by gravity after 21 days. Finally, we discovered the novel biomining microorganism, Sphingomonas dissecabilis, and provided the first demonstration of microbial interaction with extraterrestrial material on a space station in microgravity. Future investigations are needed to better understand the long-term effects of gravity and spaceflight on microbial processes, how to scale up the system, and how to keep the process sustainable. Research is needed, and it is important to highlight that this will not only benefit future extraterrestrial settlements, but also terrestrial applications. I would like to thank the Biorock and Bioasteroid teams and you all for your kind attention.
This is an executive summary of our project Oral Biofilms in Space. My name is Jeffrey Ebersole. I'm a professor at the School of Dental Medicine at the University of Las Vegas. As an overview, the Oral Biofilms in Space, or Obis project, was a collaboration between scientists and funding from Colgate-Palmolive, the University of Nevada at Las Vegas Dental and Engineering faculty, and payload implementation partner Teledyne Brown. The general concept of the project was to better understand the effects of microgravity on the characteristics of growth of oral microbial biofilms. These biofilms are present in healthy mouths throughout our lifetime, but can become disrupted, leading to dental caries or dental cavities of the tooth enamel or periodontal disease, gum disease, resulting in loss of the structures that retain the teeth. Additionally, the project assessed the capacity of existing oral health treatment agents of Colgate to work in the altered environment of the International Space Station. The general methods of the Obis process was that biological samples of healthy dental caries and periodontal disease microbiomes and saliva were collected from groups of subjects. UNLV developed a self-contained 3D printed microfluidic device to house the growth system. This oral biofilm microfluidic chamber, or OBMC, was created to house a microliter pump, batteries for power, connectors for continual fluid flow, and wells to hold 5 mm hydroxyapatite chips that mimic human teeth substrate. In each of these wells, freeze-dried onto the chips were specific pairs of saliva and oral microbiome. An important aspect of this OBMC is it enabled the flow of nutrients for the bacterial biofilms to accumulate, similar to what exists in the oral cavity. The Obis kits were composed of a variety of different pieces, primarily in order to meet all the safety and performance requirements for conduct on the International Space Station. The team constructed final Obis kits that each housed one OBMC device, two 30 ml sealed bags for the growth media, one 30 ml waste bag, and one 7 ml bag for the various fixatives to be used at the completion of each experiment and storage until return to Earth. These were all tied together with specific tubing, self-sealing lure lock connectors, and two-way stopcocks to stage the flow of fluid from each bag. The entire kit was sealed into two Ziploc bags to maintain sufficient levels of safety on the space station. Details of the method, a first step in processing each kit, required a short flow of nutrient fluid and then a 4-hour incubation to enable the bacteria to create the initial biofilm. Nutrient media flow was then initiated in each kit, and the kit incubated for 20 hours at 37 degrees centigrade in the Merlin on the space station. A second incubation at 37 degrees centigrade in the Merlin with fresh nutrient media in each kit was then conducted for an additional 20 hours. Finally, each OBMC was treated with a specific fixative agent for 30 to 45 minutes. The Obis kits were then transferred to a glove bag on the ISS. In the glove bag, the kits were separated and the OBMCs were repackaged into double Ziploc bags for refrigeration or frozen storage. The OBMCs from the space station were returned to UNLV for analysis and will be compared to identical OBMC kits run through this process on Earth. The outcomes of the study focused on 3 different issues. The first, the architecture of the biofilms that will be assessed by confocal scanning laser microscopy. The second, the microbial components, including genomics, that is, who is there, and transcriptomics, asking the question, what are they doing? And finally, the metabolome of the biofilm will be assessed, that really focuses on changes in the bacterial physiology that might occur in these microbiomes under the microgravity conditions. So that's a general overview of the project, and I will turn the next step over to Paul Galloway to give you more details on the process and outcomes and activities of the project. Thank you very much.
Good morning, everyone, and thank you so much for joining us today. This is the team from Grand Canyon University, and we are here to explain to you the photochemical and electrical effects on a microbial fuel cell in microgravity.
So a microbial fuel cell is a fuel cell that uses microbes to be able to produce electricity. We have 2 chambers. We have an anode and a cathode. On the anode side, we have the Shea-Wanella 1D sinus MR1 bacteria. On the cathode side, we have our sodium lactate substrate, which is our growth medium. So we specifically chose this specific strain of bacteria due to their high electrochemical activity. So they are able to move through this microbial fuel cell, consume the sodium lactate, and respirate off electrons. And in between the 2 sides, we have a piece of graphene felt and titanium wires, which are able to carry our electricity through.
The ultimate purpose of this microbial fuel cell is to have a ground unit and a flight unit running simultaneously. We actually just a few weeks ago partnered with the Quest Institute, and we are sending a version and a variant of our microbial fuel cell to the International Space Station on May 1st on the SpaceX CRS-25 rocket. This will give us additional data, and it will help us with additional insight of how to improve this project in the future. The ultimate space application of why this was created was in order to have sustainable space travel to distant planets such as Mars. This can use humans' waste as a growth medium in this fuel cell in order to produce electricity. As we look at the internal workings inside of this fuel cell, you can see that bacteria, which can be a form of human or organic waste, is pumped through the fuel cell, and then as it decomposes, it produces electrons. We then have the titanium wire that captures these electrons and then produces our voltage, which is our electricity, which is how our fuel cell works.
Now, for the experiment, we hypothesized that the microbial fuel cell will achieve a greater maximum output in voltage and current due to there's no shear stresses on the bacteria. Now, another cause for this is for this next launch, we also want to have a greater surface area, so that way we can get more bacteria creating more energy. So here you can see a mechanical plan of the project that will be sent up in a small module and everything that goes inside of it. And here you can see an electrical schematic of all the internal components on our PCB that fully control the project over 30 days. And in this block diagram, you can kind of see more of how everything is connected. The main part of this diagram is the analog digital converters that connect into the microcontroller. This is how we connect and capture all of our data points over 30 days. And in this flowchart, you can see how the coding program is set up on a very general scale. As you can see, we have multiple loops set up so that it is fully autonomous over the course of the project.
This whole project is based off of another launch that was done in 2019, and this was the data that was received from it. The biggest thing to note is that our hypothesis was correct. The flight unit did produce more volts than the ground unit, but we know that we're able to improve upon this output. So our previous data analysis showed that we had an 8% greater increase from 0.82 volts to 0.89 volts, which is a great increase for this microbial fuel cell. But we know that we can advance this project even further. So with our microbial fuel cell project, we want to change the potassium ferrocyanate acid catalyst to ruthenium 2,3 chloride. This makes it safer and a lot easier for us to use in space and here on Earth. We also, instead of just measuring just the voltage output, we also want to measure the current and internal resistance. By doing this, we're able to increase the surface area for these lyophilized bacteria and redesign the enclosure for our graphene felt. So our projected results, rather than just the 0.89 volts, is going to be about 1.1 volts growing into the future. We used this prior work by Gadaki, and we were able to show that this microbial fuel cell will be able to produce better with the ruthenium 2,3 chloride instead of potassium ferrocyanate and all the other technical changes that we're making to this microbial.
As we look into our conclusions after our unit is flown back down from the International Space Station, we will then collect this data and analyze it in order to create a better and more efficient ultimate variant 5 of our microbial fuel cell that we can then use in the future, and we can have different product applications here on Earth and in space. Thank you guys so much for listening. This is now the conclusion of our presentation. Have a great rest of your day.
All right, John, it's time to jump in and have a Q&A period. If our panelists would please activate their microphone, their cameras, keep your microphone muted unless you're actually speaking. That way we want to get lots of people jumping around on other people's screens. And John, take it away.
Okay, thank you so much, Alan, and thank you so much to the panelists and speakers for staying to be part of this discussion and question and answer period. We will start by looking at and discussing the questions that were provided by the question and answer feature. If anyone who is attending this session wants to submit additional questions, please go ahead. Sometimes questions are directed to one speaker. Everyone feel free to add comments that relate to your area as well. I'll try to broaden the discussion as much as possible, and we'll take it from there. There is a large question related to BioRock. I'll read it quickly, and we will start with Dr. Santomartino addressing this question to begin the discussion. As I understand, single species of bacteria have been used for each type of simulated lunar, Martian, ground, and for the control. Probably this is because there were initial studies. Could you tell us briefly what was the metabolic process, at least some, maintained by these bacteria for the mining? Which substances were uptaken, which were produced? And could microbial communities be more effective in mining than single species of bacteria? What are the limitations when choosing species? Thank you. So please go ahead, Dr. Santomartino, with some thoughts about this question.
Yes, so first of all, thank you very all for having me here and also for this question, which is very wide, but I'll try to address it. So for BioRock, we had 3 bacterial species indeed, 1 substrate, which was basalt, and 4 gravity condition. 3 of these were in space and 1 was on Earth, on the ground. And yes, I would say that BioRock and also BioAsteroid, the more recent experiment, are initial studies to test if biomining is, the principle of biomining is feasible in space, but there are so many other things that we still don't know about that and need research. And the metabolic processes, well, all the 3 microorganisms that we use, actually also those for bioasteroids, all the 4 of them, they are all heterotrophic microorganisms, which mean that in contrast to the biomining microorganisms that are normally used, which are, for instance, acidobacillus, peroxidants, antioxidants, they are autotrophic, while the organisms that we use are heterotrophic. And this has some differences in terms of metabolism. For instance, they probably perform bioleaching by producing polysaccharides and organic acids. We are studying this now. And which substances are taken, I'm not sure what this means exactly. Maybe in terms of metals that have been bioleached, we focused on rare elements and vanadium so far, but we know that also others have been extracted, and we are now studying this. And yes, microbial communities can definitely be effective. And in fact, Bioasteroid, the second space experiment, has tried to address this issue by putting 2 microorganisms together, which was 1 bacteria and 1 fungus. So try to stimulate a little artificial community. And I believe that community will be more, I don't know if more useful, but definitely useful in future for space biomining, because they could, it has been studied that they enhance biomining in some occasions, and also they can produce some organisms, some compounds that could be used for other microorganisms in the community, so they can really help each other in biomining. Oh, what are the limitations? I think the limitations are definitely all the limitations that it depends on what you mean. If you mean in terms of performing the space experiment, then the limitations are those that are often that are typical of every experiment performed on the International Space Station. In terms of going biomining on Moon, on Mars, or on asteroids, then you need to keep into account a wide range of space conditions, definitely, and also planetary protections. I hope I addressed all of them.
Yes, thank you so much,
Thank you.
Dr. Santomartino. Let's touch on some aspects of that question and see where they may or may not fit with some of the other approaches that were discussed today. Dr. Moreland, you have a very exciting technology related to containment of potentially dangerous chemicals or biologicals. And I was wondering, is your technology compatible with multiple bacterial species together, such as in a biofilm analog or in a bacterial community? And is that something that could have potential applications in your field?
Yeah, so we've actually we've got an upcoming launch actually looking at something similar. So not necessarily biofilms, but looking at two species growing together on the same plate. So previously in previous launches, the experiments have often very focused on antimicrobial specifically of different agents. And the problem we had initially was very much the fact that we tried mixing them on the ground and then keeping them cool during a launch, but we just found they mixed everywhere. And so then after that, we then had basically a long discussion trying to get this new technique approved so we can actually mix them in microgravity without breaching all the containment rules. So I think the previous ones are very much focused on testing different ones. So one experiment I developed last year was looking at honeybee venom. So that against SRS and E. coli, but we generally don't have them on the same plate. But I think biofilms is obviously quite an interesting one to try. And so I think we've got two species going up, which Halopharix mediterranei and another one, Halobacterium, I think. Not an experiment I'm developed exclusively, but those two are growing on a plate, and then we're looking at the differences in biomass and sort of how they look right next to each other. So yeah, it's more for our ones. It's more focused generally on a single species, but I think that is definitely going in that more direction, because we found initially we very much had to get our technique right to actually look at these in a very simple and cost-effective way while still following all the rules. So we love the rules.
Excellent. Looking forward to your technology. Thank you so much.
Thank you.
Let's try to take something related to that. For Dr. Huang, related in regards to the Biobits cell-free protein synthesis system. Your charts seem to imply that you could have the biochemical machinery from bacteria or from eukaryotic cells. If so, how conserved is it? Do you use one or the other? And I have other related questions to that, but in this case, what would you use for your cell-free protein synthesis machinery? And would that be something that has, again, a myriad of applications, or would have to be adapted depending on your target product?
Yeah, that's a really great question. So cell-free protein synthesis, the cell extract that is used in cell-free protein synthesis can be derived from a variety of different cells. They can be derived from bacteria cells, mammalian cells, etc. And depending on what your cell source is, that's going to determine what cellular machinery you end up with. So for example, the Biobits system is based on a E. coli bacteria extract. So it has all of the molecular machinery for transcription and translation that E. coli cells would have. So your basic RNA polymerases, ribosomes, etc. So the Biobits system is excellent at transcribing and translating proteins that are able to be expressed in a bacterial system. But you mentioned eukaryotic cells. There are limitations in terms of proteins, especially proteins that need certain post-translational modifications that prokaryotic cells wouldn't have. And so in that case, if you want to expand the applications, you might want to look into a eukaryotic-based system to make the cell extract itself. As for general Biobits applications, I talk more about that in my full-length talk. But there are a lot of different applications, right? You can use cell-free protein synthesis to generate proteins that are useful. One of the examples I mentioned in my talk is, imagine you could use the system to generate insulin on demand for if you have an astronaut with diabetes. There's also a lot of applications of using the system as a diagnostic sensor, using the proteins that are produced by this system as a visual output for different diagnostics. So you can imagine the sensor sensing things as like contaminants in water or whatnot, and then therefore triggering the sensor to transcribe and translate, say, a fluorescent protein or an enzyme that could catalyze a colorimetric output. And in fact, our Genes in Space winner from this year, her talk will specifically go over her project is actually using a biosensor in the Biobits system to be able to detect certain contaminants in water, and she uses a red fluorescent protein as the output. So in this case, we're not using the protein we're using the protein as a visual signal to know whether or not our water has certain contaminants or not.
Excellent. Thank you very much for your explanation, and looking forward to the applications, and glad to hear it has also potential applications in eukaryotic organisms with post-translational modifications. Let's ask that question. Just a general perspective to Dr. Brazil regarding your ACOR technology. Can you comment on using it for biofilms or a complex bacterial or microorganism populations, and would you have differential effects depending on the species?
Yes, so the specific 3 bacteria that we studied with Marshall Space Flight Center was because those were very good biofilm formers that's contaminating the water on the space station. But there are certainly other bacteria that contaminate that water and on the surfaces. So we know now that there are many biofilm formers also contaminating the surfaces inside the space station. And some of those are becoming resistant to the antimicrobial that's in the wipes that they're using to clean the surfaces. So yes, potentially our antimicrobial and antibacterial solutions can be used not just for water, but for other contaminants around the space station and where as needed. We have a lot of third-party validation that our treatments have broad spectrum activity against gram-positive, gram-negative bacteria and fungi as well.
Great. Thank you so much for your comments. I understand that Daniel would like to speak. Go ahead. I'm sorry, I didn't see. Go ahead.
It was just a question on the protein one, if that's all right. I was just curious how my background is in protein biochemistry, so I was wondering with cell-free protein production, there can sometimes be a problem with protein aggregation. I was wondering actually how you plan to overcome that. Were you just going to produce proteins that you know won't aggregate at high concentrations, or were you going to be a bit more broad with it? I was just wondering how you're going to try and overcome that.
Yeah, so that is currently a topic that a lot of cell-free protein synthesis experts are currently working on. A lot of the more applied applications, like actual use in the field, focus on diagnostics, right? So you don't actually need to purify the protein away, so you don't have to worry about things like high aggregation and things like that. So that's where it's moving towards right now, because yes, being able to produce proteins on demand, I didn't talk about it, but you do run into issues with protein purifications and aggregation, as you said. So there's a lot more challenges to overcome in that area. So there's a lot of work being done kind of on the preliminary aspect to try to figure out, which proteins are going to be best expressed in the system, what can we do to reduce aggregation. But a lot of the applied examples include the example we're launching next year, focuses more on diagnostics and using the proteins as a visual output. So I guess my answer is, stay tuned. There's a lot of people working on it.
Yeah, thank you. I actually work on a similar sort of experiment at the moment using urease, which is launching on CRS-24 to use a colorimetric assessment of its activity in microgravity. So something similarly related, but thank you for answering the question.
Yeah, that's awesome. Yeah, thanks for asking.
Thank you so much. Excellent discussion. Let's ask then Dr. Ebersole. Thanks again for your great presentation on oral biofilms. Can you comment on using multiple species of bacteria in relation to what may be naturally occurring as part of the microbiome of the mouth, and in relation to the tooth analogs that you're using, as well as potentially if the differential effect of capillarity in microgravity is something that may affect some of your approach or biofilm formation. If you have any comments about that.
Sure, that's a multi-part question there.
Apologies.
No, not a problem. The first thing is that we actually were using the complex biofilms right out of the mouths of the patients. So those biofilms we would predict could hold as many as 150 different species of bacteria. So the experiment was designed around actually exploring sort of real world microbiomes from the different health or disease categories. So that's part of what we're trying to study. Can't remember the second one, but the third part of it is in the oral cavity, these complex biofilms contain a lot of non-motile bacteria as well as many motile bacteria. How they will actually interact, and that's a question that we're asking, in a microgravity environment, in a non-viscous fluid environment, because the nutrients we're flowing through are just basically bacterial media. We don't know. We know that even though bacteria are only micron size, if you allow them to set in a fluid on Earth, they will settle down to the bottom of a tube. And in the oral cavity, many of these bacteria actually have cognate interactions. Receptors on one bacteria will bind to ligands on a separate one, causing the coaggregation and rapid settling of the bacteria in gravity. How that will work in the space station relative to the biofilm accumulation, accretion, and architecture are questions that we're asking. I think that answered your questions.
Yeah, very exciting research. Thank you so much for your answers.
Sure, thank you.
A related question has to do with your tooth analog. Do you expect different effects for any variability? Is that highly conserved? Some people have more or less enamel. I'm just wondering how that works. If that's also a factor that may play into the biofilm story in the oral cavity.
Sure. At least on the surface, I wouldn't think so. Within the human population, you have a range of strength and characteristics of the enamel. Some people have less enamel than others, but everyone on there, unless there's a genetic abnormality, of course, everyone has that enamel hydroxyapatite structure. Obviously, when you're using fluoride, you integrate fluoride into it, which makes the enamel molecule stronger. And helps resist the acid from the bacteria. So I think that our model with the hydroxyapatite is sufficiently consistent with the human context of enamel that I don't think we're going to see a lot of variability there.
Great, thank you so much for the information. Thanks again.
Sure.
And let's go then to Eric Yost. Great presentation. Again, the question about selecting, in your case, a bacteria that is compatible with your fuel cell goals. How many others there may be? Is it possible to use a combination of bacteria in the future, or can you touch on that as well? Thank you.
Absolutely, thank you. Yeah, that's a great question. So within our specific microbial fuel cell and all the other variants of this fuel cell that we've flown in the past, we stuck with the same bacteria of the Shaywanella 1D sinus MR1. This bacteria has been the most electrochemical active. It tends to give off the most, respirate off the most electrons that we're able to harness the most efficiently. So we have just tended to stick with those bacteria, but absolutely, if we're able to augment that process with different bacteria and we're able to find in their bacteria that is as equally or if not more electrochemically active, we can definitely put those two together and still be able to control those flow of electrons into our graphene felt and into our integrated circuit board. So yeah, that's basically the process of the fuel cell, where we're just taking the electrons respirated from the bacteria and integrating it into our circuit.
Excellent. Thanks again for the information and the great presentation. Let's go back to Dr. Santomartino. You already discussed BioRock project in terms of some of the questions. I just wanted to add something at the very end in terms of collecting the mined material. If you have any thoughts, how would that work? Would there be other steps involving other bacteria and other processes? Would you be able to recycle the bacteria after collecting the bio-mined material? Can you comment on those lines, please? Thank you.
Oh, thank you. That's interesting. As I said before, both BioRock and BioAsteroid, they are kind of initial experiments to try and understand what would be the effect of space conditions, such as, for instance, the gravity condition on the biomining process. And what we know now is that, from what we saw, is that gravity is not going to be a limit to biomining. Now, if we want to go on and understand how to scale up the system, make it feasible, etc., then there are definitely a lot of things to take into account. First of all, like for instance, the bioreactor to perform biomining is bigger than the hardware that we use in space. So you need to take this into account. And then definitely, if you want to use the metals that have been extracted from the rock, regolith, or whatever, then you need to find systems to do that. And there are, I think in this case, we could use things as similarly as they are done on Earth. So you can either precipitate them or you can use other microorganisms to do that. There are definitely other, or also you can do that by chemically. So this is a step forward that needs to be addressed in future to understand how to make the system feasible. And I think I forgot the second question. Sorry.
Yes. Thank you so much, Dr. Santamartino. Appreciate it.
Okay.
Again, the field of microbiology in space, very exciting, based on a range of impacts from the increased virulence of bacteria to recent studies on microbiome in space and its impacts, such as in the first NASA foray into human omics, as in the case of the twin study, etc., with all these other applications from fuel cells to biomining, etc. Very, very exciting experiments going on ISS. Thank you so much to the panelists for the excellent presentations and wonderful discussion. We're looking forward to your results, and I think we've reached time, Alan. If not, I was going to ask the panelists if they had any other questions for
I think we're over, John.
Thank you.
Gotta get ready for the next panel. So
Okay.
thank you guys very, very much, guys and ladies. And I was really amazed as you went through of how interrelated and interconnected all of your different projects are. We didn't try to put it together that way. It was serendipity, but it's amazing to me how they all work together. So now we're going to take a 15-minute break, actually a 14-minute break, and come back at 11:30. And our next session is on computing. So audience, go off and take a break and come back in 14 minutes at 11:30. Thank you all very much.
All right. Well, hopefully everybody enjoyed their little break there. But without further ado, let's get into our very next session. This one is on computing. Our session chair for this segment is George Frias. He's with Supernal Technical Innovations, LLC. So George, why don't you turn on your audio and video, and I'll walk us through how this is gonna go. So we'll have, George will introduce our executive summaries by name and the author, and then we will run through those 4 executive summaries. And then after that, George will come back on and we'll talk to our presenters and do some Q&A. And so we highly encourage everybody to post your questions at any time during the session, and we'll be sure to queue them up and get them asked to our presenters. So with that, George, good to see you. Well, thanks for being here, and take it away.
Yes, I'm glad everyone is attending the 2021 ISSR&D conference. This session is on computing. The first presenter will be Naheem Altaf on edge computing in space, and then Faiz Mirza, flight validating artificial intelligence software on Qualcomm Snapdragon processor on board the ISS. Then Emily Dunkel will be presenting benchmarking machine learning in the Myriad X processor on board the ISS. And then Dr. Mark Fernandez will present Advance Your Mission with onboard computing using Spaceborne Computer 2.
All right, with that, we're going to run the executive summary. So here we go. Stand by one while we get the audio going. Presentations are coming on in a second. Stand by.
Hello everyone, and welcome to our Edge Computing in Space session at the 2021 International Space Station Research and Development Conference. The solution was a great collaboration between IBM, ISS National Labs, NASA, and HPE. To begin with, I would like my esteemed colleague, Mark Fernandez, to take us through the Spaceborne Computer 2, SPC2, and then I'll deep dive into the DNA sequencing edge computer solution. Here you go, Mark. Thank you.
Hello. HPE Spaceborne Computer 2 is the greatest opportunity to easily and economically explore onboard data processing. We call that generically edge computing, and you can discover the advantages it offers using Spaceborne. Hi, I'm Mark Fernandez from Hewlett-Packard Enterprise, and I'm the principal investigator for HPE Spaceborne Computer 2. Today I want to present a brief overview of Spaceborne and hopefully get quickly to how you can use it and what you can do in terms of accelerating your research on the edge of the edge, in space, in low Earth orbit, on the ISS, and elsewhere. Pictured here are 2 identical but completely independent lockers housing HPE computer systems that make up Spaceborne 2. These lockers are designed to bolt into an express rack and contain those state-of-the-art, commercially available computers. But first, a brief history. Spaceborne 2 is a follow-on to Spaceborne Computer. This is the one-pager success story that addresses all of our SBC-1 mission objectives. First, can we package and launch a modern computer system on a rocket for installation on the space station? Yes, we can. Secondly, can the computers be installed by non-IT trained astronauts? Yes, astronaut Christina Koch from NC State, Go Wolfpack, is shown here doing just that. Finally, will those computers operate correctly, and if so, for how long? You can see that we flew for 1.8 years, well beyond our 1-year nominal mission, and we were successful on all experiments. During the mission, we focused on the computer systems and using the ISS as a data center. In retrospect, it was a proof of concept. Prior to our return to Earth, we were asked to do it again, but with a different mission, one that will be of importance to you. Spaceborne Computer 2 was launched aboard NG-15 on February 20th, 2021. Spaceborne was physically installed on April 29th, 2021 in the overhead of the Columbus module. We received full power on May 24th, taking DC directly from the ISS Express Rack. The main thing of importance to you on this slide is highlighted in green in the middle of the slide. Our mission this time with Spaceborne 2 is about you. These words are an assigned agreement and in our PIA. Spaceborne Computer 2 is about scientists and engineers advancing research. We will maintain the systems and help with the use of Spaceborne Computer 2. We have no other mission than to support your mission. And our mission is now 2 to 3 years. We are available for experiments currently underway, as well as those coming online in the next few months or even years. The hint here is to plan ahead, payload developers. Plan to process your data quickly on board and deliver the results faster than has been done in the past. So what's in Spaceborne Computer 2? Briefly, here's a summary of the hardware and software that make up HPE Spaceborne Computer 2. Spaceborne consists of 2 identical lockers, as previously mentioned, designed to bolt into an express rack as shown on the left. Other than to point out that each locker contains a computer featuring a GPU, in addition to a separate computer containing the traditional compute-focused 2-socket x86 processors, I don't want to spend a lot of detail on the hardware in order to spend more time to describe the value and opportunities of you being able to use Spaceborne Computer 2. Needless to say, SBC2 is quite capable with CPUs, GPUs, memory, and storage, all running the Linux Red Hat operating system.
Thank you, Mark, for taking us through the details. Now let's talk about what is edge computing in general. In today's world, with the proliferation of sensors, devices, wireless networks, data is being produced at an astronomical rate. To deal with the latency and bandwidth, a paradigm shift was needed. Hence, by moving data processing closer to where data is being produced to get the near real-time results and prevent losing the value of that data which is being produced, edge computing came into existence. This is especially advantageous when working in the vast expanses of space where time and energy is at a premium. It also enables nearly real-time reactions, which is invaluable in an emergency. As we know, the ISS is like a lab providing a microgravity environment for many of the experiments going on. In our collaboration, we were interested in a project where massive data was produced, and it can greatly benefit from the principles and concepts of edge computing. DNA sequencing project was selected for that matter. DNA serves as a blueprint for any organism, and the DNA sequencing is about characterizing those organisms. It can be used for monitoring these astronauts' health or diagnosing the infectious disease. This DNA sequencing project allows for the identification of microbes on board the ISS. Astronauts will take samples from the surface and run it through the DNA sequencer portable device, which is the MinION device. In initial runs, hundreds of megs of raw data was produced, and all that data had to be downlinked to Earth for further processing and eventually generating the final results. This process took over a few weeks from start to end. The objective of this project in collaboration was how we can reduce the movement of this massive data which is produced by each run of DNA sequencer from ISS to Earth, and how we can get to the results quickly for the principal investigators. Now let's take a look at the high-level architecture for this solution. In this picture, this diagram, you can see the SPC2 systems are, there's a replica on the ground, as well as we have systems on the ISS. We connected the IBM cloud to the systems on the ground. So basically, this is where the code is being developed, and the researchers, they can develop the code, which will be used to do the sequencing, DNA sequencing, and processing of the data. So there are multiple steps, as you can imagine, are involved here. So for example, if let's start, we look from the left side to the right. So in the cloud, the researchers, developers, they can go and build the code. And then that code is packaged and pushed over to the NASA facility where the HPE system, which is basically a replica of what's on the space station, is there. The code is over there, ran against the sample raw data to make sure everything works fine. And once the code is ready to go, the HPE team, then pushes that code all the way up to the space station onto the SPC2 systems. Over there, the way the process works, the main ion device on the right, you can see, the sample is taken by the astronaut or the scientist on board from the, let's say in this case, taking a sample from the surface, and it's run through the device. This device then generates close to 0.5 terabyte of raw data. Once that run is done, this raw data is accessed by the SPC2 systems and our code, which is a containerized code and running on the OpenShift code-ready container platform. It wakes up basically, and it starts going through its, there are multiple steps, which I'll take you through in the next slide. It goes through all the steps, and at the end of the run, by utilizing the two systems, SPC2 systems, which Mark explained well, that it's a combination of GPU and CPU systems, that the results are written and then downlinked to the ground system. From there, the files are shared with the principal investigator. So you can see how the developers or the scientists or researchers in a cloud, they can go and develop their code and then push it down to the ground systems, and from there it's pushed up all the way to the space station. Once the run is done, the code on the SPC2 systems shuts down cleanly. Basically, all the resources in terms of CPU, GPU are released back to the system, because the system is a multi-purpose system for many other experiments, and the results are shared in the cloud with the principal investigators. So in the end, right, like pushing the edge computing to the edge of space, this will definitely accelerate the evolution of technology back home. I mean, extreme use cases like this, they set a very high bar, and these innovations and advancements will definitely benefit edge computing on Earth as we enter the era of the 5G networks, autonomous cars, Industrial 4.0, and smarter homes. This capability, which we talked about, the edge computing in space on ISS, I believe that it will open up many venues for further spacecraft, satellites of future to perform operations in space on edge. The reducing the time it takes to get results and act on it, and help us better prepare for the future space explorations. This groundbreaking partnership, between all the partners we mentioned, will not only expedite the NASA's ISS research, but will help lay the foundation for future space explorations going back to the moon and beyond. Now just imagine, as we head towards the moon, the Artemis program, and Mars and beyond, we can use such edge computing mechanisms, devices, to detect life beyond Earth. So at the end, I would like to again acknowledge and thank everyone for their support and the great partnerships we created among all these different groups. And to all of you, thank you so much for joining the session. The information is up on the screen, and if anybody has any questions, feel free to reach out. My email address is right there. Thank you so much, and have a great conference.
I'm Fez Mirza, and today I'm going to be talking about flight validating artificial intelligence software on the Qualcomm Snapdragon processor on board the ISS. Flying autonomy software on board spacecraft can increase the autonomy. This can increase science return and decrease dependency on human operators, but it requires more computational resources than traditional spacecraft flight software, specifically CPU throughput and memory. We benchmarked several autonomy and instrument processing algorithms on the Qualcomm Snapdragon 855, a system on a chip. The primary metric is the runtime of the sample scenarios, the secondary metric being power consumption, and we run this on board a Snapdragon connected to SPC2 on the International Space Station. My full talk will talk about most of these benchmarks, however, in this brief summary, I'm going to only touch on a few of them. The Qualcomm 855 is an 8-core ARM system with 4 silver cores, 3 gold cores, and 1 gold prime core. The higher performance a core has, the more power it uses, so the gold prime core, while very high performance, is also the most costly. The 855 also has an Adreno 640 GPU, a Qualcomm Hexagon 690 DSP, a neural processing engine, and it runs Android OS. The first application that we'll talk about benchmarking was a general Fft benchmark. The two best performing processors here are the DSP overall, but the DSP only supports integers of size 8, and so when you need to use floats, which many applications do, the best performer is the GPU. The next benchmark we looked at was a matrix multiplication benchmark. And for these matrix multiplication benchmarks, we have both ARM and GPU measurements. The green and blue lines are GPU. As you can see, when the matrix size is kind of smaller, we don't get as much improvement in the GPU over the ARM processor. However, as the matrix size increases, the GPU really pulls ahead in terms of performance. And this is because there is a lot of overhead associated with running on the GPU. One of our instrument processing applications that we are benchmarking is Hyperspectral Compression, which we run on test images from Emit. The images have 640 lines, 640 samples per line, and 481 spectral bands. And for now, we only have an ARM port, but we are working on a GPU port. The measurement here is megasamples per second. That's our goal. The Emit target is 23.1 megasamples per second. As you can see in the chart, as we increase the number of cores, the runtime per core goes up, but the megasamples per second that we get goes up as well. And so for any amount of cores larger than 4, we beat our Emit target of 23.1 megasamples per second. In my full talk, I go into a lot more detail about the other applications that are being benchmarked on the 855. For now, I'll talk about some of our future work. We work to measure additional CPU performance metrics. We're currently working on getting power as well as the RAM footprint. We also intend to benchmark on additional processors such as the Leon 4, Leon 3, and the Rad 750. Any applications that are still running single-threaded, we wish to try those multi-threaded and see if we can get a runtime speed up. And we also wish to make use of the GPU, DSP, and NPU for applications that are currently only running on the ARM. And that leads me to, if you have an application that you're interested in running on the Snapdragon 855, then you should talk to us. The requirements are listed below, and my contact information is also listed here, but we're very interested in running as many things on the Snapdragon that we have on the ISS as possible. So please contact us if you have anything. I go into a little more detail about the requirements in the full talk, but they're pretty much listed here clearly. In conclusion, we ported various planning, instrument processing, and machine learning applications. We benchmarked these problems on the 855, and we're working on measuring it against other flight processors with other metrics as well. These were executed on an 855 aboard the ISS, and this work is intended to facilitate future flight of these capabilities and enable future single and networked autonomous spacecraft missions. Thank you everyone for your time.
I'm Emily Dunkel, and I and Jose Espinosa will be presenting benchmarking machine learning on a Myriad X processor on board the ISS. Future space missions will need more powerful onboard autonomy to meet mission and science objectives. For one thing, there is a time delay between Earth and space systems, and this can be prohibitive to mission goals. Deep learning is really the state of the art in computer vision, so we'd like to see if we could get these models to run on board. And this isn't something that we can obviously do due to potential power limitations and harsh space environments. So our objective is to demonstrate deployment of deep learning to a Myriad X processor on board Hewlett-Packard Enterprise's Spaceborne Computer 2 payload on board the ISS. The Myriad X processor has dedicated imaging and vision hardware accelerators. It's ideally suited for in-orbit computations due to its low thermal and power budget. During the main talk, I'll go into detail of some of the machine learning models that we have ported to the Myriad processor. These models include Mars imagery classifiers as well as Earth-based classifiers. Here is an example of a deep learning classifier that we have ported to the Myriad X. This classifier classifies orbital imagery from Mars. On the right, you can see some examples of the classes. If this model was run on board the Mars Reconnaissance Orbiter, we could do things like autonomous data collection, only downloading classes of imagery that we're interested in, as well as potentially commanding the Mars rover. If you'd like to learn more, please attend our main talk. Thank you very much.
Hello. HPE Spaceborne Computer 2 is the greatest opportunity to easily and economically explore onboard data processing. We call that generically edge computing. And Spaceborne is for you to discover the advantages edge computing offers. Hi, I'm Mark Fernandez from Hewlett-Packard Enterprise, and I'm the principal investigator for HPE Spaceborne Computer 2. In my full presentation, I provide a very brief overview of Spaceborne and hopefully get quickly to how you can use it and what we can do for you in terms of accelerating your research on the edge of the edge, in space, in low Earth orbit, on the ISS, and elsewhere. Pictured here are two identical but completely independent lockers housing HPE computer systems that make up Spaceborne 2. These lockers are designed to bolt into an express rack and contain those state-of-the-art commercially available computers. I will cover the main thing of importance to you, shown in this slide highlighted in green in the middle. Our mission this time with Spaceborne 2 is about you. These words are in our signed agreement and our PIA. Spaceborne Computer 2 is about scientists and engineers advancing research. We will maintain the systems and help with the use of Spaceborne Computer 2. We have no mission other than to support your mission. And our mission is now 2 to 3 years. We are available for our experiments currently underway, as well as those coming online in the next few months or years. The hint here is to plan ahead, PDs. Plan to process your data quickly on board and deliver the results faster than has been done in the past. I'll cover a summary of the hardware and software that make up HPE Spaceborne Computer 2. Spaceborne consists of two identical lockers, as previously mentioned, designed to bolt into an express rack. Each locker contains a computer featuring a GPU in addition to a separate computer containing the traditional compute-focused two-socket x86 processors. I will not go into details of the hardware in order to spend time to better describe the value and opportunities for you in using Spaceborne Computer 2. Needless to say, Spaceborne 2 is quite capable with CPUs, GPUs, memory, and storage running the Red Hat Linux operating system. In addition to the breadth of capabilities, I'll also cover a couple of specific examples. One demonstrates edge-to-cloud capability. From above the clouds, on the edge of the edge, in space, on the ISS, we burst down to the Azure cloud and ran classic Hello World. Many other experiments are in the planning stages to pre-process the raw data on Spaceborne, burst the extracted features to the cloud, take advantage of the cloud's large-scale computing capabilities, and return the results, the insight, the recommendations, etc., to the edge to the ISS. In another example, the results or insights from an experiment are shown to be 20,000 times smaller and downloadable in seconds. We can compute faster than we can download. In addition, in session 0-3-1, IBM presents DNA sequencing capabilities with Spaceborne. In session 0-3-2, JPL covers the radar and other instrument signal data processing done by leveraging Spaceborne 2. And in session 0-3-3, JPL further discusses image processing and feature extraction results accomplished on board. I encourage you to check out those sessions. Finally, I'll explain that Spaceborne Computer 2 is a collaboration between the ISS National Lab and HPE and available to all. The ease with which you can take advantage of SBC2 is covered. With that, I thank you for your kind attention. I wish to acknowledge our team and supporters and hope you enjoy my full presentation and the other presentations in this session.
All right, I think that does it for our executive summary presentation. So George, if you'd come on in and we'll bring up and let's have all of our presenters please turn on your video, and then George will get the discussion and the Q&A started. George.
Yes, first I'd like to thank all of the researchers for their excellent research and contributions. Let's see, I don't see any open questions. I have a few of my own here. So for Mr. Althoff, so edge computing and edge AI are topics that are gaining much attention, and there are many new conferences and events being organized around the world. So aside from this conference, which of those have you engaged in?
So, yes, thank you so much, George. And thanks everyone for joining this session. So there are many conferences, like there is a CubeSat conference, there is the cloud conferences where we have presented. Last year, we presented at the UNUSA and the United Nations in their main session. So there's like close to like 30 different conferences around edge computing and cloud, which we have presented in the last 12 months.
Thank you for that answer. Let's see now for Mr. Rosa. What radiation testing, if any, has been performed on the other new processors that are being employed here, the Snapdragon specifically?
Hi, George. Yeah, thanks for the question. We are doing radiation testing on the Snapdragon on the side. I'm personally not super aware of the results yet. We haven't released any results for that yet, but there is an element of our task that is working that angle as well.
Mm-hmm. So for Dr. Dunkel, how's the integration of the Movidius and Snapdragon processors accomplished within the SBC2? Exactly how do you do that? I know with the neural processor, it's a USB stick, right? So
yeah, so I think same here. It's just but Mark Fernandez could answer this better than I can. But It's just a stick in there.
Oh, each one of them are just a different stick? Or so you have a board that you plugged into the HVC2?
The Movidius is a USB thumb drive shaped device, and so it is plugged directly into a USB port. The Snapdragon is a small board, which we physically mounted inside Spaceborne 2, and it connects via a USB cable to an available USB port.
And also a question for you, Dr. Fernandez. See, there's a 10 gigabit interface on each SBC2. Can they exchange data with those interfaces, or?
Yes, correct. The Ethernet cables are not shown in the figures, but they are physically connected. The astronauts did physically interconnect both lockers via 10 gig E. So within Spaceborne 2, we have that high-speed connectivity taking place.
But then the downlink rate is much reduced.
Correct. We still go through the NASA network for downlinks, and Spaceborne 2 is allocated 2 megabits a second downlink.
So here's another question. How protected is cloud computing from malicious activity? How secure is this? I'll just throw it out there to anyone that wants to answer it.
So this is Naeem. So from the cloud point of view, I mean, and we are connected with the data centers on the ground at NASA, right? For the VPN through that's very highly secure with the SSL. And then I think from there, it's because the networks are not outbound, right? So that that saves information directly hitting the computers, and then there's a network between the ground and the space station itself. So it's isolated. Yes, it's not. We don't have a direct access yet.
Yes. So what you're saying is that the ISS infrastructure from the ground up is really secure, and the same thing from space to ground.
Like we call the air gap environment, right? So there is no outbound connections going from the SPC2 system. So it's highly secured. Yeah.
So what other type of research? I know, Dr. Fernandez, you had a chart that described that, but could you maybe go off a page on there? But you've gotten a lot of different customers now, it seems like. So
yeah. I personally sort of collecting them into 3 buckets, if you would. One is outside the ISS, and that is external Earth observing sensors of various types and processing of that. The second bucket is internal observations, and that includes the DNA sequencing that was mentioned. It also includes other types of imagery, for example, manufacturing images of things that have been manufactured that like to be processed, other types of internal sensor information. And the third bucket goes in the bucket of autonomous mission operations and exploring that. What sensors and gauges and other type things are on board the station that could be monitored on the station rather than monitored from ground, which would give us faster reaction times when things go into an anomaly situation.
Mm-hmm. or when you hit LOS, right? I mean,
Correct. Yeah, the LOS is, for those that haven't seen them, is quite shocking and disturbing. I often put up the LOS for the 24-hour period, and when I have bring on new partners, and I say, if your cell phone coverage was like this, you'd find a new provider. There are so many LOSs in a 24-hour period, and they can range from as little as 1 second, which is irritating, to 20 to 30 minutes. And that's when you learn that you need to be autonomous. And we have constructed Spaceborne Computer 2 to autonomously monitor itself and take actions as needed.
I don't see any more questions at this point. Do any of you have questions of each other?
I think it might be helpful to the rest of the community if these current partners gave an open and honest assessment of how easy or difficult that it is to use the Spaceborne Computer 2, and if it has in fact helped them advance their research. I have no feelings. You can't ruffle my feathers. Anything you say I will take as important feedback on how to improve the process. So please be free and let your fellow members of the space community know about this.
Yeah, so it's been really easy for us, I think. Once we had the Snapdragon installed, it's been super easy for us to basically test stuff on the test bed and then just let Mark know, oh, we're ready to do this experiment. And it's been very straightforward, I think.
And I copied that what Faiz just said. I mean, it's relatively very straightforward. Once we go and install, I mean, we just have to be careful, about the air gap. That's the restriction, which is totally understood, right? You can't just randomly go and pull information from the internet. So as long as we know that and we build our package accordingly, I think it was very straightforward. And it's actually it's a pleasure working with the marketing team.
Yeah, and I agree. It's been very, very straightforward. I don't know if maybe people would be interested in how hard is it to port the algorithms from Python or TensorFlow onto a Snapdragon or Myriad. So I could talk about that a little bit. So basically porting, so Jose did all the porting onto the Myriad, but I ported all the machine learning models I listed to a Snapdragon. And for the deep learning, for the deep learning models, it's actually, it really depends on whether you're using TensorFlow or Cafe. It depends on the platform you're using, but it's not too difficult. But Python, just pure Python, and Fez knows this, can be really hit or miss. Not all the Python libraries are supported, so you have to switch to different libraries. So we kind of go in our main presentation a little more into detail how that porting of the algorithms, just onto these processors, what that's like.
Sure, it's very challenging. How many ground units? I was going to ask, how many ground units do you have for development?
Well, we have oh, go ahead.
I encourage everyone to look at the full presentation, and in the full presentation, you see that we have what we've termed test and development systems, and those are identical hardware and software from which we give partners internet access from their home office, home data center, et cetera. And I have a personal commitment to them. If you get it running on my TDS, I will get it running on the International Space Station. And in short, we take their code from the TDS and get it up to the International Space Station. And with them looking over my shoulder via teleconference, such as you see here, they talk me through the first initial runs, and then we get into a production mode and they can begin doing their science and record time to solution, data downlink rates, et cetera, and come up with these wonderful presentations that you have today.
Looks like, oh, okay. So here's another question. I believe this question is asking about, you know, utility of using this edge computer for satellites that don't have as much compute power.
So there is one or more experiments in the proposal phase in which they plan to explore what they're calling software-defined radios. Yeah. And with software-defined radios, they can further define lower power, smaller satellites that may communicate back to a mother satellite or a mothership, which contains the high performance computing that you see on Spaceborne computer. So if your satellites do not need to communicate down to Earth, but they can communicate to a nearby super satellite or mothership, et cetera, and that mothership can do the processing and have the power to get back down to Earth, they want to explore those possibilities. And that further evolves into what's being termed swarm satellite processing, where multiple satellites can work together in concert via a Spaceborne computer too at a mother satellite or mothership to give a bigger image of whatever it is you're after to be observing.
So it'd be a cloud in the sky.
It'd be a cloud in the sky. Yep. Satellite cloud computing or satellite swarm computing.
So you don't do delay tolerant networking directly, do you?
We have that ability, yes.
Okay.
But at present, we're primarily interactive, 2 hours a week on Thursdays, until our scientists are comfortable with the processing that's taking place. And then we can run the batch processing, if you would, 7x24, 365, and get those results back to them. As you saw, the downlink of your insight is much smaller, and we can do that without disturbing our network allocation.
Yeah, bandwidth is really at a premium. Everybody wants a piece of it.
That's correct.
Mm-hmm. Looks like we've got one more minute. Let's see if there's another question. Yeah, it looks like there is. So it says, you said there was 2 megabits downlink plus LOS. Isn't it acceptable for a terrestrial ISP? Anything being worked on to improve this? That's where the delay tolerant networking comes in, but then Is it really just 2 megabit downlink?
Yeah, correct. That's our allocation.
That's your
2
allocation.
megabits per second. Yes. The aggregate is a little bit higher than that, but it's nowhere near the 1,000 megabits I have at my house. So.
Yeah. Someday.
Someday. Mhm.
So just a quick question on that. Hi everybody, it's Jim Way jumping back in again. So, what kind of technology might be coming down the pipe that might be able to increase that bandwidth, that speed? Laser comms, anything else?
That's beyond me. That's more of a NASA comms question. All right.
Gotcha.
I've got the edge computing aspect squared away, and I can support whatever network speeds you can bring to me, and I'll appreciate any and all of those megabits.
Scan test bed, maybe.
Right. There you go. Well, George, unless there's anything else, anybody have any final comments, or just leave it open? George?
I think we're
all right.
Yeah.
Well, good. Well, thank you very, very much, everybody. Excellent stuff. We really appreciate your being here, being a part of the event. It's been great. And so with that, we're going to conclude this session. Thank you again to George and our panelists. And we're now going to take a break. East Coast for lunch, West Coast for breakfast, and our Asia for a midnight snack. So please do come back at 1:15 p.m. Eastern time for our next session, which will deal with physical sciences and materials development. So yeah, come on back at 1:15 Eastern, and we'll see y'all then. Thanks very much.
Thank you all.
Thank you.
Bye.
Bye.
Hey, Boris, can you please turn off your video? We're not ready for your session yet. And your audio. Thanks. Mute yourself.
All right, welcome back to the ISS Research and Development Technical Sessions. We are in the afternoon session of the first day of sessions, of 3 days of sessions. And our session now is going to be, let me just pull it back up so I don't butcher the name as I tend to do sometimes. Physical Sciences and Materials Development 1. We have 2 sessions on Physical Sciences and Materials Development, one at 1:15 and one at 2:30. Each one lasts an hour. And one of the most important things for us is as we go through this session at any time, you can go down to the bottom of your screen and click on the Q&A and enter a question for our panelists. So please start that early and let's bury them in questions if we can. Our chair for this session today is George Frias. George, please take over and tell us who we have on the panel and what they're going to speak about.
Thank you, Alan. So today we'll have presentations from Dr. Dan Lang, carbon nanotube-based low reflectivity coatings for space applications. Boris Kusid, building colloidal crystals under microgravity. Manish Kumar, understanding the local structure part PDSNs, solders in terrestrial versus microgravity environments. And then Serena Seshardi, deepening of multi-phase fluid in microgravity using light and photoresponsive surfactants.
Thank you, George. And now we're going to play these 4 executive summaries back to back. And when we come back in, George will take over and manage the Q&A to each of the presenters. And again, please go down to the Q&A bullet, click on that, and send us some questions. All right, Kelsey or Abigail, please go ahead and run our session.
Hello, everyone. I'm Dan Wang from Faraday Technology. Today, I will talk about our work about carbon nanotube-based low reflectivity coatings for space applications. This program was funded by NASA SBIR program, and we have our subcontractor from aerospace and physical science. Future NASA missions require low reflectivity surface to minimize the scattered light for the optical systems. And the low reflective surface also need to withstand the harsh space environment with little impact on their adhesion and optical properties in broad spectral range from visible to near region. Recently, NASA has discovered the CNT-based materials can absorb more than 99% of the light, which provides the opportunity to develop carbon nanotube-based low reflectivity surface. And this is the objective of this program, to develop a scalable process for fabrication of low reflective carbon nanotube-based coatings can withstand the harsh space environment for NASA or other commercial users. The technical approach of this program is combine the electrophoretic deposition process with Faraday process to apply the functional black coatings to specific parts. And after making coatings, we characterize the coatings through optical properties. We also evaluate the properties under simulated lens conditions. We also investigate the atomic erosion and UV radiation effect on the coatings, showing as following slide. First, here is showing we coated the carbon nanotube on different shaped substrate, including the cylinder, the sharp edge, the flat substrate, also the internal and external surface of the square tube. After making the tube, we calculate the properties. First, showing the optical properties. It's showing on the top curves, the blue and the red curve is from CNT samples. We can see it's showing around 0.5 to 1% of the reflectance across the whole visible to near region. As comparison, the Z306 is the black curve, showing the reflectance is from 3.5 to 8%. The bottom showing the BRDF measurement. The Z06 pen showing the 0.01 BRTF in scatter. The CNT only have about 0.002 BRTF in scatter, which is much lower than the Z06 black pen. The simulated lunch vibration test showing here, we applied the frequency from 20 to 2000 Hertz in both directions. And after the vibration test, there are no weight loss observed. And the optical measurement showing in the red curve, there are no optical degradation and also no particle separation from the surface. We ran 5,000 hours UV illumination on the samples. On the CNT samples with the control samples Z06 pens. The results showing in the curve in the figure C. We can see the bottom two lines, the blue and the gray line, and the gray lines from CNT. So they only have a minimum 0.14 to 0.32% increase. The Z06 pens have like 2.4 to 4% increase, showing in the red and the yellow curve. The atomic oxygen erosion test over 4 days, equivalent to 41 days of the RAM-facing exposure at LEO. The results showed the mass loss of CNT samples is only 21% of Kapton and 62% of the Z06. The oxygen, the reaction probability for CNT is about 4.8. The Z06 is 7.7. HOPD is 11%, which means the CNT coatings is more resistant to the LEO oxygen erosion. In summary, we coated CNT on different shapes of substrate, and the skin coating has less than 1% reflectivity, and they can resist the UV radiation, atomic oxygen erosion. And thank you for your attention.
We consider building colloidal crystals under microgravity. I will give this presentation on behalf of a team of researchers from New Jersey Institute of Technology, New York University, University Space Research Association, and Streamline Automation. We present results of our recent experiments conducted on colloids of spherical and ellipsoidal particles on the International Space Station. This work was supported by the NASA Advanced Colloid Experiments Program and funded by several NSF cases and NASA grants. Of course, the first question is why we do need to study colloids in microgravity. The reason is that on Earth, gravitational sedimentation confirms most colloidal systems to 2 dimensions with numerous artifacts due to the presence of floors and boundaries. Unique long-range duration microgravity environment on the space station adds another dimension to the problem, making available 3D motion, 3D processing, 3D fundamental science, and 3D materials and devices. Hot sphere is a basic model of particles in the statistical mechanical theory of fluids and solids as they mimic the extremely strong repulsion that atoms and spherical molecules experience at very close distances. The hot sphere model is a cornerstone of our understanding of liquid crystallization. However, due to gravitational setting of particles, terrestrial experiments fail to confirm the crystalline phases predicted by computer simulations. The hot ellipsoid model provides a generic model for the quasi-universal structure and dynamics of liquid crystals. Compared to molecules in liquid crystals, which are too small to be seen with an optical microscope, the colloidal particles are huge in size and can be used to test predictions of computer simulation. And the conclusions of our talk is that up until now, the theory of the simplest most common, most fundamental crystallization transition from liquid to face-centered cubic FCC crystal had not been confirmed. Now it is confirmed. We demonstrate that simulations of equilibrium phase transitions work only when gravitational effects are unimportant. Our results validate that computer simulations of equilibrium material structure can be used in microgravity. In more details, prospects to employ microgravity to address grand challenges in soft matter science and applications are summarized in the recent NASA publication listed on this slide. Thank you.
Hello everyone, this is Manish Kumar, second year PhD student at ISSR&D AIMS. My presentation is focused on structure and properties of solder joints produced in terrestrial and microgravity conditions. This project is funded by NASA PSI, and I would like to acknowledge Dr. Richard Grugel and Dr. Louis Stranzenberg, a NASA scientist, and Dr. Ralph Napolitano, professor at ISSR&D AIMS. For space-based fabrication and repairs, which will be essential for future long-duration human exploration mission beyond low Earth orbits such as Moon and Mars, it is important to advance our current electronics and mechanical joining capabilities in space. So in this presentation, I will discuss how soldering in microgravity is expected to be different as compared to that it is in terrestrial conditions. In microgravity, there is lack of buoyancy force, and the dominating forces are due to surface tension. If we compare the shape of solder ball produced in terrestrial and microgravity conditions in image A and B respectively, the solder ball is hanging downward in the direction of gravity, whereas it is in football shape in the microgravity conditions. This football shape is due to the dominating surface tension forces. We can also see the cross-section image of terrestrial and microgravity solder in the image C and D respectively. The amount of porosity is higher in the case of microgravity solder. This increased amount is due to the lack of buoyancy force and the reduced fluid motion in the absence of gravity. This increased porosity may affect the mechanical integrity as well as the thermal and electrical conductivity of the solder joint. Now I will discuss some results from the microstructure characterization and nanomechanical testing of terrestrial and microgravity solder sample. As you can see the schematic of terrestrial sample in the top left, the gravity direction is downward, the buoyancy force is acting in the upward direction, and the solidification is in the downward direction because nucleation is starting at the copper wire. As we can see in the backscattered SCM images, there is inhomogeneous distribution of lattice phase and tendrils phase throughout the cross section, and we also see that there is the amount of lattice phase is increasing when we move in the direction of gravity because lead is the heavier phase. Also, we can see some amount of porosities at the solder joint. Now I will discuss the microstructure of the microgravity solder sample. As we can see in the backscattered SEM images in the top right and the bottom right, there is homogeneous distribution of lattice phase and tendrils phase because there is no gravity effect. We also see some porosities at the solder joint, and which is higher in the amount as compared to terrestrial solder. We can also see in the top right image that there is some material smearing that happened during the mechanical polishing in between the voids, and it could be possible that the solder structure is weaker in between the voids as it is away from the voids. Now moving on to the microstructure and porosity distribution comparison in microgravity and terrestrial solder. As we can see in the backscattered SEM images, the microstructure of microgravity solder is coarser as compared to terrestrial solder, and it could be possible that there is a large number of porosity in the microgravity solder, which reduces the heat transfer and it further reduces the cooling rate, and because of the cooling rate, the microstructure is coarser. Also, if you compare the porosities in the bottom left graph, the amount of voids in the microgravity solder is 51 across 4 cross sections, which is almost 6 times higher as compared to terrestrial solder. Also, the voids in the microgravity solder, as we can see in the bar graph, are distributed over a long diameter range, whereas in terrestrial solder these are distributed over a short diameter range. Now moving on to the nanomechanical testing of the microgravity and tested soda sample. As we can see in the bar graph in the left hand side, which shows the hardness of different phases measured away from the voids. The hardness of lead-rich phase, tin-rich phase, and from the composite response of lead-rich phase and tin-rich phase is lower in case of microgravity solder as compared to tested solder, and this lower hardness is believed to be due to the coarser eutectic structure in the microgravity solder. This is our future research plan. We will finish the microstructure characterization across remaining cross-section. We will also perform high-resolution microcomputed tomography to see the distribution of 3D distribution of porosities and soda microstructure. We will also perform nanomechanical testing to see the effect of intermetallic layer on the solder strength and the spherical nano indentation to obtain the nano indentation stress strain curve. We will study the effect of cryo and elevated temperature on the solder strength and the intermetallic layer growth using the in situ luminous nano indenter. With that, I will end my presentation. Let me know if there is any question or suggestions. Thank you.
Hi everyone, my name is Serena Seshadri and I am presenting my work on depending of multi-phase fluid in microgravity using photoresponsive surfactants. This work was done at the University of California, Santa Barbara, across the chemistry, chemical engineering, and mechanical engineering departments. Thank you for having me today. The motivation for this work is to enhance boiling in microgravity environments. Boiling is important for thermal management, power generation, water harvesting, amongst many other applications. But on Earth is well understood and occurs over 4 regimes depending on superheat. In microgravity, we want to maintain boiling as close to the critical heat flux as possible, and in the nucleate boiling regime. Here, bubble dynamics play a significant role, and nucleation growth and departure strongly affect the boiling performance and ability to reach high critical heat flux. Bubble departure relies heavily on buoyancy. In microgravity, in the absence of buoyancy, the efficiency is decreased. So we seek an alternative approach to move bubbles in the absence of gravity. We utilize the photomarangoni effect for this work. Similarly to the Marangoni effect, it relies on the surface tension gradients that lead to surface flow, simply established by light, giving the name photomarangoni. In this study, we introduced photoresponsive surfactants to establish surface tension gradients and promote bubble and droplet departure from solid surfaces. Photoresponsive surfactants can reversibly switch their molecular conformation and therefore change the interfacial tension of solvents when illuminated with the light of selected band of wavelengths. This makes them exciting candidates for fluid manipulation because light can be easily reconfigured with high spatial resolution and instantaneous response. An ideal surfactant to achieve bubble departure in a multi-phase system should display a large surface tension change, good reversibility, fast switching kinetics activated by low-intensity light. As such, we selected the spiropyrin-based photoisomerizable molecule, which we call here SPDA-PEG. The surfactant design enables the largest change in polarity upon photoswitching, as the initially amphiphilic molecule loses its hydrophobicity upon irradiation. The isomerization of the spiropyrin isomer to the mericyanine isomer is activated by UV light within 300 to 375 nanometers in wavelength. And the reverse isomerization occurs upon the removal of UV irradiation, which can be further accelerated by application of 500 to 600 nanometer light. We see here large changes in surface tension and interfacial tension of the spiropyrin in solution against gas as well as in toluene against water. This is observed at low light intensities and with a high degree of fatigue resistance, with rapid kinetics as well as rapid changes in interfacial tension, making the spiropyrin an ideal candidate for both control over organic and aqueous solutions. We carried out experiments on the departure of a liquid droplet consisting of toluene containing 0.1 millimolar spiropeg in water. The experimental setup is shown here where a toluene droplet is injected into a water bath through a round hole using a syringe pump to control the injection rate as well as the volume, which leads to changes in droplet departure time. We apply a UV laser light to the top of the droplet in order to drive fast bubble departure. And here we can see that using low light intensity, the droplet departure is realized in less than 0.4 seconds and can be controlled by changing droplet volume or location of light. We further conduct finite element simulations in order to investigate the lift force due to the photomarangoni effect, as well as to understand the location of the force that drives the bubble departure. We find that surface tension change of 29.4% generates flow both within and outside of the droplet and is enough to enable force for the bubble to depart in microgravity environments.
All right, George, that's the end of it. And we apologize to the audience. Yes, we know we had a very bad echo on that, but we didn't have any open mics and it was purely within the system. There was nothing we could do to reduce that. We tried several things. So George, if all of your panel would turn on their cameras and go through Q&A, George, and your discussions, please.
Thank you. Well, it looks like we have a couple of questions here that have popped up. The first one here says, presumably the electrophoretic deposition technique could be adapted to other carbon nanomaterials, e.g. graphene or chemically reduced graphene oxide, etc. What are the limitations on the substrate? So who would want to take that question? It looks like Boris is, well, go ahead.
I think it's a question for my talk. Here's Dan Wang from Fft Technology. The answer is yes, other carbon materials can also be used for electrodeposition, but first you need the carbon materials dispersed in solvent, so forming the electrolyte. Second, you need some surface charge of the materials. So then when you apply an electric field, so the charged particle can move and deposit on the target substrate when you apply substrate. And the limitation of substrate, since they are using the electrophoretic deposition, so the substrate have to be conducting. So we have successfully deposited the carbon materials on like stainless steel, aluminum, beryllium, ITO, all of these conducting substrate. Yes, that's the answers.
Okay, thank you for that answer. I have a question for you as well.
Yeah.
So we're all used to knowing that, say, a car with a black interior gets hotter than a car with a white interior. So could you explain the thermal characteristics of the CNT coating while it's in direct sunlight versus when there's no sunlight at all?
You mean the hardness of the materials, or I didn't get the question. When the carbon material comparing with what materials?
When it is in direct sunlight, what is the thermal profile of that?
Okay. We didn't measure the thermal properties, but the nanocarbon materials have very high thermal conductivity. So they also used in some interface materials for the thermal control or thermal control systems.
Mm-hmm.
I don't have like direct data of this part thermal measurement.
But it wouldn't be as well. It absorbs the heat, right? But
they absorb actually they have a very wide band spectral band to can absorb the heat from like UV region to mid IR region. They can absorb all of this spectrum, so they can use like absorb all of this light and heat.
Okay, but would that make it hot then?
Yeah, and I think then I need to transfer the heat to other target.
Okay. All right, thank you.
Mhm.
So for Ms. Seshadri, so is it conceivable that the method you are using could be used to aid boiling fluid in space? As far as I know you had experimental setup there that had certain type of fluid, but could it be used for any type of fluid?
Yeah, so ideally, this photosurfactant that we are designing and studying is soluble and switchable in multiple solvents. And so we chose toluene because it is the best studied system for our kinetics and everything. However, this spiropyrin is also effective in water, and so therefore can be used for boiling. We're working on some studies with the bubble departure in strictly water, but it can be used with multiple fluid phases as well. So that's why we study with toluene in water, but also this can be used with any other organic and aqueous combination. So ideally, yes.
What about the edibility? I mean, if you're making soup.
Yeah, I'm not sure about that component because it's not very, I wouldn't consider this an edible surfactant, at least in its current form. I'm sure we could edit the synthesis and full structure, but at this point, not yet.
Yeah, I noticed that the gentleman had the soldering presentation, talked about the Marangoni effect, but since this would have to be added to the whatever it is, it wouldn't really apply. There wouldn't be any way to apply that there.
Right, it would have to be added into one of the fluid phases, or if in the case of boiling, to the water.
Thank you. So this is for Mr. Kossid. In your last chart, you describe how you plan to build for the future. Could you describe an application for the equilibrium materials you propose? What type of, usage would there be for that? So are you muted, Boris?
I didn't hear very well your question, George.
Okay, so you were talking about the plan to build for the future on your last chart.
Yes.
Yeah.
We did experiments on colloidal systems of spherical and elliptical particles. And this is monosized colloids and very high concentrated. And it's in the presentation, it's given all details. And what is important that in colloidal or spherical particles, we observed formation of a large crystal FCC, which about 2.6 centimeters, which exactly was predicted many, many years ago and has never been observed in terrestrial experiments due to glass transition or something what people didn't understand, and still not clear why terrestrial experiments are different. But it's quite clear that gravitational settling substantially works, even if we try to match density as much as we can. Because actually we never can match density. We are talking about micrometer size gaps and many days of experiment. That's why this is what happens, and that's the answer. And with elliptical particles, we also confirm formation of pneumatic phase, what was predicted and has never been observed, because you cannot do experiments on elliptical particles. Due to sedimentation, they form 2D structures. And for the first time, we were able to see 3D structures. That's the basic difference.
That looks like a breakthrough there. That's awesome.
Yes.
Incredible. But what you described as equilibrium materials looked extremely fascinating as well, in that they were in a state of without any energy. Is that correct?
Yes, this is just pure systems which were well predicted and can be studied by molecular dynamic simulation, because it's just entropy-driven phase transitions. It depends only on the concentration, and that's it. If you get any attraction forces or whatever you get it in the system, then it's very difficult to predict, because that's what not quite clear, maybe simulated. And also in our presentation, we showed a slide where periodic tables and what we are learning in all undergraduate or even high school chemistry classes, that most metals form FCC or sometimes HCP structure, majority. But why is that? Actually, it explains that, yes, because there is a much better arrangement, but nobody was able to observe it. And you cannot, simulations predict it, but it was not observed, because we never can do on atoms, we cannot see atoms. When we do simulations of real atomic system, it's very difficult to predict the repulsion or attraction forces. And that's why colloidal systems started from the beginning of the 20th century, from the Nobel Prize of Perrin, who use them to model atomic and molecular systems, because that's ideal hot spheres, and there is only limitation that they are not impermeable. It's not so easy to fabricate them. You need to eliminate all kind of interactions, get coated with some hydrocarbons, and to prevent any electrostatic forces. And also you need to get fluoropore, but fluoropore you cannot place on the surface, because it's immediately generate zeta potential electrophoresis. So you need to get fluoropore inside the particle. That was an art fabricated in New York University by Dr. Hollingworth. He is extremely magician, and he was able to do it, and he was able to fabricate spherical particles, and then these particles were embedded into film, stretched for several days, again wash out, and then you get monocytes elliptical particles. So it's very easy to say to do those experiments when particles are available, but you cannot buy them.
Thank you for that explanation. It's really fascinating work. Mr. Kumar, I've done a lot of soldering in my day, and I know a cold solder joint, it may look okay, but it really is not. It won't conduct properly. So does the increased porosity of the solder joints in microgravity make it impractical to use conventional ground-based methods on orbit?
Can you repeat last line?
Pardon?
Can you repeat last line? What was the last line?
So does the increased porosity of the solder joints in microgravity make it impractical to use conventional ground-based methods on orbit, ground-based methods of solder?
So I don't think we can use ground-based solder method, because these samples were prepared about International Space Station. Same method was used as ground-based samples, and like in both the comparison of terrestrial and microgravity solder, we see the higher amount of porosity in microgravity solder. So I think somehow we have to improve our soldering method to decrease the porosity.
Yes, that's, I think, very valuable research you're doing. So are you, for in the future, are you going to advance and try to come up with a better method for zero gravity soldering?
Yes, so recently, like maybe in next 1 year, we are planning to do some new experiments for International Space Station, and we are using Marvin instrument designed by TMI INSEE, TechMaster INSEE, and using that instrument, we will prepare new samples and we will see how we can improve the soldering, and we are will use some method to remove the porosities from the solder joints. So yeah.
Looks like we have some other questions here. So can you use DEP on uncharged particles?
That's for the Fourier deposition, or?
It's not specified.
Let me answer that question. For the uncharged particle, if they can be polarized under the electric field, then they still can use the PED, Fourier electrodeposition. So after polarization, the particle can move and deposit on the substrate. But if cannot polarize it and also have no surface charge, then cannot use the PED approach.
So this next one is the CNT single wall, multi-wall, semiconducting or conducting.
So for the data I showed, we use a multi-word carbon nanotubes, but we also coated the black coating with single word carbon nanotubes. Both work for this method, but considering the cost, so we use multi-word carbon nanotubes for larger area coatings. So the semiconducting conducting, like we also coated like graphene oxide, kind of like an isolator. So that's for the conducting or non-conducting isolating semiconducting is not affect the EPT approach.
Here's another one. Are there any challenges with expansion or contraction of the CNT materials possible over a larger application?
For the expansion, so let's say first we have to make it's produced CNT. It's produced CNT like a bundled powder. So first we have to well disperse the carbon nanotube in solvent. So after the coating, we show like 2 by 2 inch square tubing. We didn't see any construction or expansion of the coatings. We also have other like 4 by 4 inch coatings. Didn't see this challenge. We have not tried a larger size of the coatings.
Okay, so this is for the crystal experiment for microgravity crystal formation. They tested mutated, genetically synthesized polypeptides to be crystallized further in microgravity. So what are the details of that study? So did you get that, Boris?
I cannot say anything. I know that some experiments on proteins were conducted, some of them conducted by pharmaceutical companies, and they don't share results openly. And moreover, whatever was shared, it's quite difficult to interpret because the systems are very complicated. And that's not a lot of theoretical predictions. We intentionally worked on extremely simple system where theoretical predictions are available. Otherwise, very difficult to understand what you see. For example, we did boiling, And we published a paper, and microgravity, boiling of water, bubbles survives up to 280 Celsius degree. It's very strange, but it happens. As our paper was published in NPG Microgravity. Somebody may take a look. Yeah, so microgravity is not so simple. You don't know what you see, and then it's very difficult to interpret what you see. So because the time experiments is limited and efforts is a huge, it's multi-million dollars exercise. It should be very well conceived experiments for which everything is predicted, where it's clear understood that there is some difference what is expected and what we see in terrestrial experiments, and then it makes sense to move to microgravity. We ran experiments for about a year on microgravity. Different sets, and I have been running experiments on airplane. It's very, very difficult to interpretation. If you want to do something fundamental, you need to get a very good selection of your experiments and clear understand that all artifacts would be not critical, that you can interpret them, because from 2D structures, from anything we see on the ground, we unexpectedly get 3D structure. And by the way, most simulations cannot predict gravity effect, because that's from the very beginning, gravity, any phase transition, any process under gravity, it's not equilibrium. There is a motion, which we don't have, even very small motion, micrometer scale. But if you are talking about half a year of time, it's a huge displacement. So that's why the microgravity is the only real experiments where you really get equilibrium, can reach equilibrium. Then you can compare with MD simulation. You cannot do it for terrestrial. Sometimes it's only if you're lucky and your gravity effects somehow do not affect what you do. But you don't know in advance what gravity affect, because gravity generates motion. As soon as you get motion, it's not equilibrium. To which extent your simulations are correct, you have no idea, because you cannot do non-equilibrium still on huge number of particles. It's not feasible, even on supercomputers, especially crystallization. More or less boiling, you can predict vapor bubble is small, but when you get crystallization, it's very small change in chemical potential. It needs to be a huge number of particles to get a sizable change in free energy. But you get a sizable change, then you cannot run MD simulations.
Okay, thank you for that. This question, I believe, is for Serena. So in the last talk, does the Marangoni convection also happen due to thermal gradients induced by the light source?
Yeah, so that's definitely a great question. Something we considered quite a lot. So in our simulations, we calculated that in that amount of time that it takes for those droplets to depart, which is less than 0.4 seconds, we only expect a 0.25 degrees Celsius change in temperature. And experimentally, with the highest possible light intensity, which was 44 milliwatts per millimeter squared, we only got about a 1.5 degree Celsius change. So we expect that the thermal Marangoni effect is negligible. Some of the other experiments that we tried to confirm that, we found that to induce a similar surface tension gradient, we needed upwards of 5 degrees Celsius. But with our experiments, we never get temperature gradients that are large enough to induce that kind of a thermal Marangoni flow. So we expect that it's negligible. I'm sure it has some effect, but not overpowering the light driven.
So now the last question here for Mr. Kumar. Cold welling out in space, should this lead us to understand more about materials?
So
I'm not sure if this question is related to soldering because both are different techniques. Like welding is cold welding is different, where we are just trying to join the two blocks through maybe like friction welding or something. But in soldering, we are melting the metal and then we're solidifying. But I think cold welding may help us to understand more about the materials in microgravity. But I don't think you'll see there any such effect of the sedimentation or something like that.
Hey, thank you for that.
So I have one question to Serena, if I can ask.
Sure.
So have you seen any relationship between your bubble departure and its size? And can your simulation be used in the metallic fluid?
Yeah, so we have definitely seen a relationship with size. So naturally with the larger droplets and bubbles, it's quicker to depart and it relies less on the location of the light as well as the amount of time that we supply light to it. And so with smaller droplets and bubbles, we require that the location of the light matters significantly, and that's what our simulations were about. And so it definitely depends on the size of the droplets, but we're able to reduce those sizes by having the light. And what was the second part of your question was if we could apply?
Can we extend this to the metallic fluids?
I'm not entirely sure if our surfactant system would be stable in metallic fluids. It's definitely something worth considering. I've never really tried or thought about that deeply, but I would love to think about that.
Well, I think we're at the end of our time. Is that correct, Alan?
Not sure if Alan's there, so I'll jump in here for a second. No, you actually have until, I believe, 2:15. So you got about 10 more minutes or so.
It's just 15 minutes every.
Just a fan. Thanks, Jim. Yeah, I couldn't find my button. I've lost my button.
Okay, we can keep going. So do any of you have other questions of the panel?
So again, I would like to ask Serena, like you mentioned, when bubble size is increasing, we are seeing the earlier departure or easy departure. So is the surface tension affect the departure there? Because I expect when the bubble size is increasing, the surface tension will decrease around the void, both sides of the voids. So is that the reason, like the possible forces to remove the bubble decreases when the size increases, and that causes the easy departure? Or is there some other reason to that?
I believe that for our case, since these experiments have just been done on Earth, so these aren't in microgravity conditions just yet, when the bubbles are much larger, they depart due to buoyancy at these larger droplet sizes and volumes. And so when you have smaller droplets, we have a larger pinning force on the bubble and droplet itself. And so we require that surface tension gradient to be able to drive the departure rather than relying on some buoyant forces, which is relevant in the higher volume and sizes. So that's the main reason. Certainly the surface tension gradient changes as the droplet size increases as well, but mostly it's due to buoyancy.
Yeah, because in microgravity, we can't expect the buoyancy force to help in the departure. So is there some other reason that like causes the departure? So that's why I was asking.
I have a new question. Is ultrasonic welding the same as cold welding?
So I'm not exactly sure about the ultrasonic welding, like how is it done? So I'm not sure.
Let's see, what ways can early entrepreneurs get their ideas for ISSR&D studies? So that's not relevant for this panel. So.
George, let me throw one in here to Dan. Dan, you talked about being able to do the nanotube deposition to affect good thicknesses and reflectivities or absorption of radiant energy. Two questions on that. The first one is, are you doing this kind of research on orbit also so that you understand how it works on orbit? And second one is, a lot of people are beginning to look at how to manufacture large satellites and large optical systems in space. Would your system be applicable to that kind of manufacture?
Okay, first question, we did the coating just on the ground, but we did a simulated land condition test, basically like a web test. We also did like the 5,000 hours UV illumination to simulate the space environmental. And also we investigate the atomic oxygen erosion in the simulated LEO environmental. We did all of this kind of simulated environmental testing for the coatings, but we have not sent the samples to space for the measurement. Is this for your first question?
So that answers it. Thank you very much, Dan.
Mm-hmm.
And one for Boris. Boris, you were talking about the crystalline structure and fixing the crystalline structure in microgravity. You talked about colloids and different kinds of materials. Is it just casual to is it obvious to the casual observer that this is also applicable to metals, or is that another step?
From the very beginning, colloids was introduced to study phase transition because it's quite a number of reviews shows that it's the same as metals. But the big difference that when you do experiments on real atoms or molecules, you cannot see at the level of a single particle because you have to either go to Brookhaven lab or to get some a neutron beam or something. You cannot watch it. It's very fast and particles not visible. Advantages of colloids that they are micrometer size and you can watch and record everything at real time at the single particle level. And it was first introduced by Perrin when he measured gas constant with this in 1900 something. Because colloids are the same. Thermal brotion is there, and they are nicely model what's going on with metals, of course, simple metals. We are talking not about very sophisticated crystalline structures, but simple metals, that's the same. If you take a look at periodic table, you will find that most of them form FCC, because the basic feature of all liquids and solids, the first issue, that they are permeable. And only after that, there is some difference between different materials due to attraction or repulsion variation. But the first step, it's impermeability. And that colloids have been used for many, many years as an ability to provide ability to monitor on real time frame at the particle level. That's what exactly I presented, just a couple of basic results because we finished experiments just in June, beginning of July. but we measured dynamics of particles. And it was very interesting results, which was not expected. And again, simulations and dissimulations cannot capture it without any empirical fittings. When you get empirical fittings for forces, you don't know what you have. On the other hand, when you get gravitational force, then it works. And unfortunately, this, when you are talking about micrometer size materials, films or something, and if you get displacement 0.01 micrometer, just 1 nanometer a second, then multiply by a month, then it's a big displacement. So it's not equilibrium. And and we still don't know how to barely handle equilibrium phase transitions in materials. And within equilibrium, it's very difficult. Because again, we need to get a large number of particles, which is not affordable even for supercomputers.
Thank you, Boris. I appreciate it.
Yes.
Go ahead, George. Close us out.
Okay, I want to thank each of you again for your hard work in this research. And Alan, I would like to address that one question about how to get involved with the ISS conference, and that would be to go to astronautical.org and keep track of when the call for papers comes out and look at the call for papers, the requirements, and submit a paper. So you got anything to add to that?
No, that's a great summary. I wish we had thought of that. It was super of you, George. Thank you.
Yeah. Okay. Y'all have a great day. Thank you for coming to the conference.
So thank you all for being in this session. Thank you very much. And now we're going to take a break until 2:30 when part 2 of materials and in space is going to be presented by George again. Come back in at 2:30.
Thank you. Bye-bye.
unmute. I have to unmute myself. Welcome back, everybody. Welcome back. Hopefully you're enjoying these tech sessions so far. It's great to have you being a part of the ISSR&D conference. We really appreciate y'all being here. Once again, my name is Jim Way. I'm Executive Director of AAS. And let's just keep right on going, get right into our second session on physical sciences and materials development. Our session chair for this one is once again, George Frias with Supernal Technical Innovations. So George, I'll turn it over to you to introduce our executive summaries, and then we'll roll the tape.
Thanks, Jim. So this afternoon we'll have another group of very interesting research papers presented. First, we'll have Richard Weber presenting microgravity characterization of molten metal oxides and synthesis of novel glasses. Then Howard Warner, low Earth orbit fabrication and characterization of zero-length Z-bland optical fiber. Then Stephen Wilkie presenting levitation and microgravity helps link thermophysical properties with atomic structure in molten oxides and glasses. And then Lori Chang presenting space test of integrated photonic devices. And finally, Prayanti Amarasinghe, Eucurious halide crystals growing in International Space Station. So Jim, if you want to roll those.
All right, we'll get the presentation started here in a second.
Hi, I'm Rick Weber. I'm going to be talking to you about microgravity characterization of molten metal oxides and synthesis of novel glasses. This is a presentation that's in the session 05, talk ID 17. And this work relates to measurements of thermophysical properties of glass-forming metal oxides that are important in a number of applications. And the goals of the work are to measure the density, thermal expansion coefficient, viscosity, and surface tension of the molten oxide materials, including in the supercooled state as they move towards the glass transition. Results will be disseminated through publications, presentations, and through NASA's PSI database. And we're going to use the thermophysical property data obtained in this research along with atomic structure measurements that are made using X-rays and neutrons on both liquids and glasses to increase the understanding of glass formation. The flight experiments will be performed on ISS using the JAXA ELF instrument, and some of the samples are actually being run this month in August, so we have some preliminary data that wasn't available in time to put in the recording, but it's now being analyzed. Some compositions form glass relatively well in containerless conditions. The ELF is a non-contact processing method. It doesn't touch the sample at all. Sample's held in position using electrostatic forces. So one can supercool liquids and maintain high chemical purity even at temperatures above 2000 degrees centigrade, where some of the materials of interest melt. We've measured the atomic structure of a number of these materials in ground-based work using the Advanced Photon Source and the Sporlation Neutron Source at Argonne and Oak Ridge National Labs, respectively. And one of the goals of the analysis and writing up and processing the results will be to compare property and structure data to get additional insight into the structural evolution and property evolution that occur side by side as one moves towards the glass vitreous state. So just a brief summary of the experimental sort of protocol. Samples we're looking at are made of things like lanthanum titanate and barium titanate, calcium aluminate, some aluminosilicates, and some magnesium silicates. Quite a range of fragilities and melt behavior expected among that set of materials. We have around 60 minutes per orbit between cutouts in the connection with the ground. So a typical experimental run might run around 60 minutes, or possibly the sample would be cooled and remelted. So the sample's inserted, heated up, and then melted by heating with a laser beam. And then we bring it down, hold it in isothermal steps, measure the surface tension and viscosity by drop oscillation as we approach down into the supercooled region here. Then the sample's solidified, heated back up, and then cooled, during which time density is measured by imaging the sample to determine its. So in summary, the experimental plan is developed. The first batch of samples are being run this month. Structure data has been measured on most of these materials on the ground, and we're expecting to get data back and be analyzing it as we speak. So we look forward to combining this with the structure data. So thank you for your attention, and we look forward to questions and discussion.
Hello, I am Howard Warner, Director of Advanced Developments at Mercury Systems Torrance, and I will be discussing Mercury's Orbital Fiber Optic Production Module project, known as ORFOM, and its associated fiber optic production systems, known as FOPs. Last year, I presented ORFOM at the ISS Research and Development Conference and introduced the FOPs that Mercury is developing for the production of high-performance Z-band optical fibers in the microgravity environment of low Earth orbit. Z-band optical fiber produced in space has the potential for performance characteristics that greatly outperform today's state-of-the-art optical fibers. Producing Z-band fiber that has twice the bandwidth and 1/100th of the attenuation loss of modern-day optical fibers can greatly advance the development of fiber optic based technologies such as high-powered lasers, networks, medical devices, and military defense applications. Since that presentation last year, the team at Mercury launched the second FOP system, known as FOP 1.5, for operations on the ISS and conducted several zero-gravity experiments that have advanced the technology and influenced design changes to take us several steps closer to producing the world's highest performing multi-mode optical fibers. The first of those zero-gravity experiments occurred shortly after last year's ISS Research and Development Conference. The team at Mercury had the opportunity to conduct several parabolic flight experiments to validate a new design for automatic initiation of the fiber draw process and to evaluate the efficacy of applying a nitrogen purge to the drawing process to reduce the extrinsic impurities that form on the fiber as it cools. The results were successful, and the lessons learned from each were incorporated into the most recent FOP design. A month after the parabolic flights, the team assembled to coordinate the FOP 1.5 experiments aboard the ISS. Over the course of a week, Mercury conducted 15 experiments in 3 separate groups that resulted in the production of nearly 100 meters of Z-band optical fiber. Each successive experiment and each successive series built upon the lessons learned from the previous experiments. With some aggressive iterative adjustments, the team was able to hit an objective of drawing over 20 meters of optical fiber from one preform. This was a first ever milestone for space-based production of optical fibers. Post-mission analysis of the optical fiber back on Earth confirmed that the last few experiments were able to produce sufficient quality fiber to compare with similar fiber that was produced on Earth. These results demonstrated that space-based production of Z-bland fiber in a less than ideal environment still produced fiber that performed as well as fiber that was produced on Earth in an inert gas environment. The lessons learned and experience gained as part of the parabolic flight and FOP 1.5 experiments have heavily influenced the design of Mercury's third FOP system, known as FOP2. With the addition of an advanced heater design, a low-volume nitrogen purge subsystem, an automated fiber initiation subsystem, and an improved control algorithm, FOP2 has great potential to produce vastly more fiber of higher quality than any other previous system. Mercury is now at the midpoint of its FOP development strategy and is heading towards the development of a nearly fully automated FOP system that will be capable of producing commercially viable Z-bland optical fiber in low Earth orbit. At Mercury, we keep the world connected at the speed of light by making optical fibers that are truly out of this world.
Hello, I'm Stephen Wilkie, and I'll be briefly summarizing our work using levitation and microgravity to help link thermophysical properties with atomic structure in molten oxides and glasses. We're interested in thermophysical properties because they're both technologically relevant and also help advance our fundamental knowledge of materials. On the technological side, any manufacturing process that deals with the liquid phase depends strongly on thermophysical properties such as density, viscosity, and surface tension. For example, in additive manufacturing, such as selective laser melting, you have a laser beam where you're melting a powdered bed and obtaining a local melt pool. And the fluid behavior of this melt pool has a strong effect on how the part turns out. And so these properties will be very important for advancing in situ resource utilization where we're making parts in space. On the more fundamental side, much of our research deals with glassy and disordered materials, and there are still many unanswered questions about the nature of the glass transition. One particular question is what underlies the distinction of different liquids into so-called strong and fragile liquids whose temperature dependence of viscosity is different. Strong liquids have a clear exponential dependence on inverse temperature as they cool, and fragile liquids have a strongly non-Arrhenian temperature dependence for their viscosity. And we're interested in exploring what results in these different types of liquids, and can we relate that back to the atomic structure of the materials? And so the main goals of this project are to explore connections such as that between atomic structure and thermophysical properties. We are measuring thermophysical properties on board the ISS using the electrostatic levitation furnace, or ELF, that is built and operated by the Japanese Aerospace Exploration Agency. And with that, we are obtaining density, viscosity, and surface tension on a variety of different oxides. And if you're interested in more details, please check out my colleague Rick Weber's presentation about this aspect of our project. I instead will be focusing today on our complementary measurements we're doing on the ground, looking into the atomic structure using X-ray and neutron diffraction, with a specific focus on one glass composition, neodymium titanate glass. And for our structural analysis, we utilize pair distribution functions, which essentially describe the correlations of atomic pairs in the material. The reason that we use X-rays and neutrons for diffraction is because these measurements provide complementary contrast to the different atom pairs. And this is because X-rays are scatter more strongly off of heavier elements, whereas neutrons don't have this dependence on atomic number. And in the neodymium titanate glass, we have 6 unique pairs of atom types. And so the neutrons are going to be more sensitive to lighter elements like oxygen, whereas X-rays are more sensitive to the heavier elements. Shown here on the right are 2 PDFs, or pair distribution functions, obtained from our experiments for the X-ray and a neutron measurement using a isotope substitution for neodymium. And as you can see, the weighting for the pair correlations is very different between the two samples. For example, the oxygen-oxygen is much weaker in the X-ray measurement than it is in the neutron. However, these plots are jumping to the punchline a bit too soon because the experiments themselves only give us the total PDFs. And there are a lot of challenges from a single measurement analyzing this due to overlapping peaks, which is why we've obtained an X-ray measurement and 5 unique neutron measurements via isotope substitution. We've taken these 6 unique diffraction measurements and fed them into an empirical potential structure refinement model, which uses an empirical potential to drive the model towards structural arrangements that better describe the experimental data, which is in good agreement shown here between the experiment in orange and the model in blue for each of those 6 measurements. From that, we can analyze the pair correlations and deduce bond distances as well as atomic coordinations that match well with prior studies and give a much more complete picture of the glass. And the structural analysis lays the foundation for our upcoming thermophysical property measurements and dynamic studies. Thank you. I look forward to your questions in the live Q&A.
Hello everyone, my name is Lori Chen. I'm from University of Delaware. My major is electrical and computer engineering. Thank you for inviting me to participate in the 10th Annual International Space Station Research and Development Conference. Today, my topic is space tests of integrated photonic devices. Thank you for Drummond and WhatsApp Lee, who are my partner to help me accomplish the project, and also my advisor, Dr. Gu. Please go to the next slide. Here are the collaborators and members who provide the photonic integrated circuit and space test. I appreciate their contributions so we can have an amazing project and result. Please go to the next slide. We relied on communication devices such as working on projects, reaching out to friends, etc. Those figures from the upper left corner to the upper right corner show the transition of the communication and navigation system since 1998. And the speed of the RF communication we can see from 2016 to 2019 is getting faster. Compared to the optical communication, it is the most fastest communication device so far. So communication technology has become a frequent and important tool in the nowadays. Please go to the next slide. Here are the Rocket-C program launch. In our group, we have sent several chips to the space station by a rocket on June 22, 2018. There are many devices on the chip, including micro-ring resonator, Mach-Zehnder modulator, etc. I will explain more details of the function of in the full briefing PowerPoint. Please go to the next slide. Vacation from International Space Station and how they flown to the space and where they stay in the space. To test this function, we manufactured silicon photonic devices that were flown on the International Space Station and were mounted externally on the ISS. With nearly 6,700 near-Earth orbits to ensure the device's exposure time to the Van Allen radiation belt, which is full of energetic charged particles and ionizing radiation. Those chips are staying in the space nearly 6 months. Please go to the next slide. This shows that where the chips are and how they are packed on the rocket. You can see that we have 5 chips sent to the space on the right corner here. Please go to the next slide. Here are the images that were taken after exposure. We will use these after-flight chips to compare with the performance of the pre-flight chips so we can see the difference between them. Thanks to the UD nanofabrication facility, so we can use the scanning electron microscope to have a nice picture like lower right corner. Here we can see the straight long rectangle upper right corner has a MZM devices on it, and the image below that, the square one, has a microring resonator photonic crystal waveguide and meta surfaces. We will explain how ionizing radiation affect the device after the testing. Please go to the next slide. For your listening. I appreciate your time. And if you are interested in this topic, please go to a technical session from August 3rd to the 5th. And you can also ask me questions on August 16th at Q&A section. Thank you very much.
Hello everyone, my name is Priyanti Amarasinghe. My other team members are Jusu Kim, Sudhir Trivedi and James Jensen. This presentation is on Mercurius halide crystals growth in International Space Station. If you look at the properties of Mercurius halides, the crystals grow in positive uniaxial tetragonal crystal system. They have a very high birefringence. They also have a very high transmission range from UV through long wavelength infrared region. They also have high acousto-optic figure of merit. Therefore, these materials are excellent material for acousto-optic applications. The material is very dense and very good for radiation detection application. Before growing the crystals, we need to purify the material. However, purifying is very difficult because of the low solubility of the materials in most solvents. So we need to purify the materials by sublimation. You can see black impurities remaining in the already purified ampule. We do purification few times to get rid of all the impurities before we start growing the crystals. Then the purified material is crushed and placed in the growth ampule. And we place the ampule in a crystal growing furnace system. Mercurius halides are grown using physical vapor transport method under congruent evaporation. During the growth process, we maintain a temperature gradient between the source material and the growing crystal. After the crystals are fully grown, we start the cooling process. After the crystals are taken out from the furnace, we characterize the grown crystals using X-ray diffraction technique. Mercurius halides have prominent cleavage plane along 110 crystallographic direction. Because of that, the orienting the crystals are not very difficult. To check the crystalline quality, we measure the full width at half maximum of the rocking curve. One of the best applications of Mercurius halides is using it in acousto-optic device fabrication. Here at BIMROS, we are researching on using the materials for acousto-optic tunable filter fabrication, mostly for long wavelength infrared region. This schematic shows the basic principle of acousto-optic operation. You have the AO crystal, which is cut and polished to the correct orientation, and a transducer is attached to the acoustic cut of the crystal with an appropriate RF frequency. The transducer is producing an acoustic wave. The optical beam interacts with the acoustic beam and gets diffracted. Using this technology, you can filter or separate different wavelengths from the incoming optical beam. So this technology can be used in applications such as hyperspectral imaging and point detection. Part of this work includes growing Mercurius halides on ISS. The idea is when you grow crystals under Earth's gravitational force, a buoyancy-driven convection flow is generated. This convection flow may affect the crystalline quality of the crystals. At this point, we are working on the simulations of crystal growth under microgravity conditions. Once we gather all the parameters using simulations, we can modify our crystal growing technology so it can be used in experiments on ISS.
Hi, I'm Rick Weber. I'm going to be talking to you.
All right, that does it for our executive summary presentations. So it's now time to start discussion and Q&A with our panel. So George and all of our panel, please turn on your video and mute unless you're talking, please. So if everybody could come on in, there we go, Richard Weber. And we're looking for Lori and Howard to come on, and Priyanthi as well. So here we go. All right, with that, George, take it away. There's a couple of questions in the Q&A. I will tell everybody else, please submit your questions using the Q&A tool. All these people are here for you to answer your questions, and it's some really pretty cool stuff. I pulled Howard's ear, you know, a week and a half ago or so, just talking about his stuff, because it's just wildly interesting. So ask away and let's talk about it. Take it away, George.
Many congratulations to you all for your successful research. Your successes, and looking forward to talking to you about them. Let's see if we got any questions yet. We do have a couple of questions. Let's look here. How exactly does microgravity affect the structure of glasses? How are they different from those produced on Earth? More flexible, porous, pressure resistant, ductile, and why? That's a mouthful.
Maybe.
Sure.
Yeah. Hi, this is Rick Weber. I guess I could take a stab at that one if you like. So is the audio working okay?
Yes, I hear you very well.
Yeah. Good. Okay. We're actually using microgravity to achieve control of the melt so that we can measure the thermophysical properties as the liquid cools down to form a glass. And the reasons we might see different structures in the glasses that are recovered is mostly due to differences in heat transfer that would affect the fictive temperature and consequently small changes in the behavior of the materials, the refractive index and density perhaps. But at this stage, we're not so much concerned with production of the glasses in space as characterizing the liquids as they cool towards a glass, and then combining that with atomic structure data in order to understand the evolution of structure and properties. And in fact, Stephen Wilkie commented on that in connection with fragile liquids. And that's really where we're sort of focused in this research.
Excellent. Thank you for that answer. Let's see. I have some questions myself here.
I would be curious too if Howard has anything to add to that. I know we had a couple of glass talks ranging from atomic structure up to pulling fiber. So that last question might have also been looking more at the macro scale with those Z-bland fibers.
Yeah, I can certainly add a little bit to that. I mean, from our perspective, the whole hypothesis of drawing optical fibers in a zero gravity environment is explicitly to address the formation of crystallization in those heavy or exotic glass structures as they go through their phase transition of being heated up into that taffy, like pulling environment. When we pull that fiber, as it exits the heat flux in the heater, and it exhibits a rapid cool down, much like any other crystalline structures, they got to get aligned somehow. And so what we see on Earth is that in the presence of gravity, there tends to be, to some degree, extrinsic impurities through crystallization and other things that occur. So what we're trying to prove is that without gravity, we can minimize that crystallization. Now, with respect to the pliability, the strength, I don't think it's going to be much of an impact on glass in particular, at least the glass we're dealing with. But for us, it's pretty exciting, right? I mean, how often do you get to isolate gravity from a manufacturing process? So, so far so good, and we're looking ahead at, as I mentioned in the presentation, I think that the low Earth orbit zero gravity environment will have a positive impact on not just glass, but several materials as they go through phase transformation and get to the final condition that we're looking for. And so what those differences are being determined. I think that's probably about all I have to add.
Anyone else have something to say about that previous question?
I'll just add one other comment that to build on what Rick said too. I don't expect there to be large changes in regards to atomic structure just for being in space versus on Earth. However, if you have a glass that phase separates, which is very common, into two different liquid phases, those most typically have a density difference. And so any sort of transport process driven by buoyancy-driven convection or anything else is going to shape those phases potentially in a different way in space than you would see on Earth. So for things that have multi-component, or sorry, multi-phase, I think you might see a difference.
Excellent, thank you. So here's a question, Howard. Why do you have only 60-minute windows? Are you a potential user of the HP edge computing? That was a previous session that we had before lunch. They can do the processing in that it's like a supercomputer and reduce downlink time. But I'm not sure if you're familiar with that.
Was that question directed at me?
Yes.
Oh yeah, I'm okay. I'm grasping at straws right now because I could you repeat the question, because I'm not sure what the 6-minute window is.
60-minute window. Yeah, as far as you have a 60-minute window every orbit, right?
Oh, so yes, but that's mostly driven by the comms windows and the orbital mechanics of the International Space Station. So really, I mean, in the experiments that we conducted back in December, there were several experiments that we were all teed up, ready to go, but because of the handover from one comms link to another, just because, you know, we need at that point for that particular design, we needed to send data signals up to the machine as it was, the FOP machine as it was drawing. So that 60 minutes is approximate. Sometimes it was 45, but what I would say is that as you look ahead in the, you know, in that presentation that I provided in the executive summary, there was that V curve, and as we look ahead to a presence in low Earth orbit, whether that's via a short duration up and down, such as a Dream Chaser platform, or a permanent presence, such as a low Earth orbit space station, those limitations will no longer apply, right? Because it'll be fully automated. It'll really just be a matter of replacing the material. So to my knowledge, and again, I'm not aware of any other limitations. Our only limitations that were 60 minutes or less were based on the orbital mechanics and the comms windows associated with the ISS as we were actively sending signals to and from our FOP machines.
Yes, I believe that the edge computer could do that automation for you now, possibly, but that's a different question. So I appreciate your answer. So the next question, do crystals grow faster in microgravity, and is the shape growth path the same? Here one sees it grow according to its confinement. Thank you. So who wants to take that one? Basically, do crystals grow faster in microgravity? Okay, so we'll go to the next one. This is directed to Stephen. Are you doing your X-ray and neutron crystallography on orbit or after you return to Earth?
That's a good question. Definitely after we return to Earth. The specific conditions that we need to do the scattering techniques on amorphous materials like these glasses require both very high flux and also, in the case of X-rays, very high energy. And so that means that we do our X-ray measurements at a synchrotron source, at the Advanced Photon Source in Illinois. And then the neutron measurements, again, to get the high flux that we need, we go to the spallation neutron source at Oak Ridge. And our plan so far, so the results that I discussed today were on glasses that were made here on Earth, and we're waiting to get samples back from space after they're tested on board the levitator there. And then we'll do diffraction measurements on those as well and be able to compare directly glasses that were made on Earth and in space.
Excellent, thank you. So for Mr. Weber, so could you explain the practical applications that may result from your research? Something that we would see an improvement in our lives? Or...
We're looking at fundamental studies on glass that would be used in models. So accelerating development of applied glasses is really one of the areas that we could bring to bear on that. For example, in the case of some of the display glasses that are used in cell phones, a lot of modeling's been used in those to develop them. And one of the things that's missing is often property data and benchmark data that can really help to improve those applications or models of applications. So I think that's really where we're going to see some advances.
So I assume it's somewhat unpredictable, but it's really great research you're doing. Thank you.
Yeah, I mean, it's an underpinning. There's a lot of discussion at NASA at the moment about transformative research, and I think the position I have on that is often that you need to do fundamental research to be well prepared to understand phenomena that emerge. And then when applications are being developed, you have that fundamental knowledge to apply and really accelerate the breakthroughs that are needed to bring new technologies out.
Mm-hmm. So there's another question in regards to the last talk. Were the radiation sources considered and or characterized as part of the experiment?
I believe this is my topic. So I think the radiation source is considered as a categorized as part of the experiment because the space has like different, like also really already exceed the radiation and like heavy proton or something like that. So we just want to narrow down like which source like affect our devices or like lattice structure.
Thank you.
Does it make sense? Thank you.
So for Howard, this question asks, how do you initiate the draw without gravity pulling the fiber down? Well, if you're in microgravity,
Yeah.
there is no gravity.
Yeah, that's right.
So unlike hanging a conch shell upside down and hoping gravity pulls the creature out over time, in zero gravity, we see the cohesive properties of the glass help keep it, keep its shape, keep its volume. Actually, before I continue, I want to go back to that previous question that while we'd be certainly happy to entertain HP edge computing, I would probably get in trouble if I didn't explore Mercury's edge computing first. So if that doesn't work out, then maybe we can talk some more. But thanks for that question. For this particular question, so the drawing process, whether it's in gravity or in microgravity, essentially remains the same. You have to, there has to be something that initiates from the end of the fiber optic preform or the glass rod. And that initiation can literally be anything from touching it with another glass rod to, what we did in our first few experiments with the previous two fiber optic production machines was to just essentially tie Teflon tape around the end and just pull a giant bait mass out of there and then try to manage the mass flow rate in and out of that heat flux. What we did as part of those 0G parabolic flights and what we demonstrated and proved was that during those, that 20 to 24 second window of zero gravity with a very responsive heater, we saw that the glass was pretty much maintained its volume and its shape. And then when we initiated the fiber draw with our process, which was not a large glass rod, but something a lot smaller to be able to get in there and maintain the initial contact with the fiber optic preform, and then have enough friction to be able to pull that out. As long as you can meet those requirements, what we found was that the fiber initiation was much more predictable and much more reliable. So again, independent of gravity, but I will caveat this by saying here on Earth, we do have a second machine that is a twin of what is sent up to the International Space Station. So with respect to gravity, we do in fact rotate that machine on its side so that, at least we get the gravity vector going in the right direction. And that does help us. That way we don't, get the soft glass sticking to the side of any walls or anything like that. But up in the space station and on the 0G parabolic flight on that 727, it works like a champ.
Excellent. Here's a general question. How do you measure radiation in space? I don't know if that's exactly relevant to any of your papers.
Yeah, so we don't measure radiation in our space. We just know there's a radiation might affect our devices. So we just want to make sure how does it affect the devices.
Mm-hmm. Yeah, it's the number of rad hits, and it can affect the computers and everything. So.
Yeah. So, but if you want to know more detail about how radiation affects the device, you can go to my full briefing slides, like slide 7, and you can read some paper about this.
Okay, thank you. So Howard, how might we expect to see our lives change on Earth because of the breakthrough you have with the fibers?
Yeah, so
I know you talked about some things, but it's not really in those terms, I think.
Yeah, no. So, in the longer presentation, there is a brief discussion on potential applications. But look, I'm a former submarine captain, right? So I spent a lot of time enclosed underwater, not up in space. But what I do understand about fiber optics and the practical application therein, it doesn't, in my mind, it doesn't take a large leap of faith to understand that if we can deliver more energy, whether that's in the form of data or in the form of energy to energy, right, to apply high-powered lasers or defensive weapon systems or whatever the case may be, if we can create the conduit to do that with an exotic glass fiber such as ZBLAN or any other combination of fluorinated glasses, as I mentioned in the longer presentation, this could fundamentally transform the fiber optic industry in itself, because now we're providing a material that had otherwise not existed, that allowed a much greater transmission. In the case of data, if you know, the theory, right, is that we can get twice the bandwidth in the near infrared to infrared spectrum, and we can get 2 orders of magnitude reduction in attenuation loss. So starting with that reduction in attenuation loss, what does that mean for the optical fiber infrastructure? What it means is you need less hardware, things like amplifiers, you know, so the costs associated with building that infrastructure would go down. That would be a benefit in many ways. And then, you know, certainly from the perspective of twice the bandwidth, you know, using, you know, the process of wavelength multiplexing, you know, you can essentially, you know, last year I brought up the analogy of the robot on the new version of Lost in Space, right? The digital robot that was sort of a cross between organic material and metal. And, you know, what comes to my mind is there's probably a single fiber optic cord, like a central nerve, running down that machine. And, you know, if we can get multi-mode fibers that are able to carry, you know, the data or a signal from a single complex processor that tells it to do, you know, multiple different things, much like a human body. You know, we see something, we want to reach for it. Our mind is telling our hand to move in that direction, to make an intercept, to grasp it, pick it up and do something with it. And we ought to be able to, you know, apply that kind of logic and reasoning through the advancement of artificial intelligence applications. So, you know, I'm not, again, an expert in the predicting what AI or any increase in data or energy will bring us down the road. But I do think transforming the optical fiber industry and the infrastructure and its applications, given how optical fibers are ubiquitous in our daily lives, from short transmission to long transmission, I think the potential is rather large. And I do think we can see an increase in several applications, whether it is in the medical domain, whether it is decreasing the latency of machines like the Da Vinci machine that allows doctors to operate from afar, or whether it's developing, particularly in our domain, Department of Defense applications that may help save the lives of not just our service members, but, you know, those out in the battlefield. So, and again, it's limitless, but those are probably some good discussion points to explore further. So I'll stop there.
So it says theoretically it will increase up to 2 orders of magnitude. That's just through that one pipe. So there's another question: how will ZBLAN provide double or more the data if we're limited by erbium amplifiers and other telecom C-band equipment? Well, that's a limitation of that equipment, not the new technology. Is that right?
Yeah, I think I understand the question. That, and you know, I'll backtrack and say that again, there is a current state of the art. There is an existing state of the art in the optical fiber industry. But if we provide these companies and these innovators in this industry with a new pipeline of optical fibers that can outperform state of the art today by orders of magnitude, and, you know, either in bandwidth or in attenuation loss, I think it is reasonable to assume that the supporting infrastructure equipment will also need to be upgraded, and there would have to be some evolutionary processes there. And again, I don't pretend to know what that would be or how much that would cost, or, you know, over the long run, but I think, you know, the basic logic train is more information and more energy, you know, through existing pipelines of information or energy. I'm sure we will find something positive to do with that. And so, you know, I think that what we have seen from the interest of potential commercial customers, you know, are things like increasing the transaction speeds of whether it's financial exchange rates or whether it is just sharing information at a university between a wide area network and being able to, again, provide more information with less infrastructure. So I think there are several opportunities there, and our business development team is, we're trying to get them to go down the path of doing homework on that, and they're doing a pretty good job of identifying these potential markets and down the road. And again, I will say that this is all contingent upon the results of our upcoming experiments, where we have almost achieved everything that needs to be achieved: an inert environment, zero gravity, and a consistent diameter. And if those fibers come back and show this type of qualitative improvement, because the math works, right? The math tells you this is what is supposed to happen if you can pull a fiber of this purity. I think that we'll have to be a little bit smarter then about where we go with this and how it's applied.
So here's a question for all. How do you move what you are each learning in space? How do you move that down to the Earth? For example, how do we learn how to manufacture Z-blend on Earth?
I'll answer the Z-blend. So humankind, already manufacture Z-blend on Earth. In fact, Z-blend as an exotic glass has been around for decades. It is a formulation of exotic glass, of fluorinated glasses that have been explored and experimented with for a very long time. And I would say probably almost 40 years now. The challenge and the reason that Z-blend or fluorinated glass fibers aren't applicable is because of that, the several different compounds and the inherent crystallizations that occur in the gravitational field. Earlier there was a discussion that perhaps, and it's a small effect, but in the short duration time between phase changes for Z-blend glass, there may be some, for lack of a better term, sedimentation of the heavier molecules over lighter ones. But again, every little bit counts. And so I think that the practical application is that we know how to do Z-blend on Earth. In fact, there are several companies that do it. It's just that the performance quality of that fiber does not warrant a commercial product at this point in time. So that's that. And then I'll answer the second question real quick, and I'm sure everyone else wants to chime in. But as far as how to do the same thing on Earth, I would just end by saying that is our whole hypothesis, is that you cannot do it on Earth, and we have to do this in low Earth orbit. So we're very thankful for this opportunity to conduct these experiments to prove that.
I'd be willing to throw in a few comments. I think one of the values of the microgravity research in the area we're involved with is to benchmark materials and understand the mechanisms of why they're better. And then that gives clues as to how they can be improved on the ground. It doesn't mean one necessarily will reach that benchmark in every case, but if you understand the mechanism that leads to the improved performance, then you've got a basis for designing a process differently from how it's done now. Maybe modifying flow with different kinds of orifices, for example, if you're pulling fibers, or in the case of large-scale glass production, perhaps mixing the materials differently or controlling temperature gradients differently. So it's, again, going back to an earlier comment, it's sort of an underpinning for technology, for technological advancement.
So anyone else have thoughts on that? How to bring it to the Earth? I mean, how would you manufacture it on the Earth? Take what you learn and apply it here.
I would say for the thermophysical property measurements that Rick and I are looking at, similar to what Howard said, these are very uniquely enabled by the microgravity environment, because you can get such more stable levitation, and the quality of the data is much greater. The variety of materials that you can levitate and get good quality data on. far surpasses that of ground facilities, which do exist. The same sort of electrostatic levitators are, there's one run by NASA at NASA Marshall, and also a ground-based one in Japan. But it's expected that the success rate for different types of samples will always be surpassed by the measurements done in space.
So Lori or Yanti?
So I think our project goal is to use these devices in the space. So we don't, I think we want to just know how it changes and we can modify it on the Earth. And after sending it to space, it has like same performance or better. Yeah, that's our goal.
Yes. One question here, how much of a difference will the atomic behavior of fiber optics exhibit when manufactured in 0.17 G versus 0.38 G?
Yeah, so great question, right? Because, one of the things that we did on our parabolic flights was we did, in fact, start out at moon gravity and then the, gravity equivalent to that of Mars. Actually, the other way around. We started out with Mars, which is about 0.3738, and then lunar gravity being about 1/6 of that of Earth. We did that as well, though. Those experiments that we did on the parabolic flight were strictly limited to the auto initiation process and the efficacy of nitrogen on the quality of the on the quality of the fiber. I think, I don't know if it would be a linear relationship. We don't know, but it's certainly something that once we establish our moon base alpha, we can, I would love to go up there and do some experiments, no doubt about it. My guess is there will be a impact negatively on fiber optics with this particular exotic glass fiber, given the more gravity vector you provide. And so I do think there will be some negative impact on that. But again, how much that is yet to be determined. I think if we could bookend the results, and I think what Mr. Weber had said earlier about establishing baselines and looking at what you can explore from that and understanding what changes you can make. Right now we're establishing bookends. We know what happens in 1G, and we think we're going to find out what happens in 0G. And so something in between there is about where lunar-based and Mars-based manufacturing will fall.
Thank you very much. So do you have some questions amongst yourselves you'd like to ask? Got what, 2 minutes, is it?
Yep. Any last words? All right. Well, hearing none, thank you very, very much, everybody. George and our panel, thank you so much for being here, for being a part of this. It was a great discussion and some really exciting things to look forward to. So thanks again, everybody. Appreciate it. All right, we're now going to take a 15-minute break, 15-ish minute break. We'll be back at 3:45 p.m. Eastern time. We'll be right here in the same Zoom room for our last panel of the day on Earth science and remote sensing. So stay tuned, and we'll be back in about 15 minutes. Thanks, everybody.
Thank you. Thank you. Thank you. Thank you. Whoa. All right, that should be much better.
Okay, and Brian, it's Jim Way. Just mute yourself and turn off your video because we are actually still streaming live. We just don't turn it off between sessions. So just come on, turn on your video when the session, when the executive summaries are done, and mute unless you're speaking.
Good afternoon and welcome back everybody to the final session of the day for the ISS Research and Development Conference technical sessions. This session today, this afternoon, is Earth Science and Remote Sensing. Kenton Fisher of NASA is our chairman for today. And I'd like to first start by reminding everybody to go to the Q&A box on the bottom of your screen, click that box and start putting in questions as soon as you start developing them. The more you get in, the more will get answered. So Kenton, would you please introduce your topics and your presenters?
Thanks, Alan. So everybody, my name is Kenton Fisher. For those not familiar with me, I am the principal investigator of the Crew Earth Observations Facility for the ISS. That is all of the crew imagery the astronauts take for research purposes. And so today we've got a nice set of talks today from 4 authors. First one is going to be on measuring carbon dioxide from the ISS using the OCO3 mission by Anne-Marie Eldering. We've got the ICM-90, which has got to do with these high-resolution multispectral CubeSat constellations by Raphael. And then we've got the SAGE-3 instrument being presented by David Flitner. And then finally, we've got a session on the video from the International Space Station by Brian Walls. And with that, I'll hand it over for the talks.
There we go. Thank you, Kenton, for that introduction. And now we're going to play the executive summaries of those presentations end to end. And we'll come back after that to get the panelists online. So Abigail, go ahead and play the sessions, please.
Hi, everyone. Thank you for joining for my presentation on measuring carbon dioxide from the International Space Station. I'll be talking about OCO3. Our mission is aimed at measurements of carbon dioxide in SIF. We were operational since August 2019. And in a presentation, I'm going to share information about our new mode called SAMS that we collect, shown on the upper right here, and also on some of the new data we're releasing called B10 with improved geolocation and improved consistency across the mission. We are located on the GEM-EF. OCO3's payload is here, sort of facing in the flight direction on the GEM-EF. And we're called OCO3 because there was an OCO2 that is a currently operating free flyer. OCO3 is the same spectrometer packaged for the GEM-EF. We collect measurements of reflected sunlight in 3 bands, the A band, which is sensitive to clouds, and then 2 carbon dioxide bands. These are used in a retrieval process to make estimates of carbon dioxide. And very importantly, we have a ground-based measurement system, FTS is called TCON, that are tied back to the internationally recognized standards for carbon dioxide as part of our validation program. These SAMs I mentioned are a unique capability. Because of the pointing mirror system that we have, we can collect data over 80 kilometers by 80 kilometers in just 2 minutes. More details in my full presentation. With our 2 by 2 kilometer footprints, this gives us a really great map of carbon dioxide, and we do this over cities, power plants, field campaigns. We collect between none and 30-something per day over many different types of sites. For many locations in the world, since the mission started, we've got over 24 measurements. Some of these are cloudy, but we've really developing a deep set of data. Just an eye chart to show you some of that data. Los Angeles measurements here on the left and Toronto on the right. Los Angeles has very clear enhancements due to it being of a large city with air trapped by the mountains and prevailing winds, often onshore. Toronto is less obvious about the enhancements over the city, less terrain, more variability in winds perhaps. So rich data set that we do need to continue and investigate. This movie just highlights that data collected over Los Angeles. And the lower panel shows you a time series of the concentration. So you see that the background's about 414 parts per million. And over the areas of enhancement, we see up to 419 parts per million of carbon dioxide. We've got a new data set that's coming out now, just started in July. Improved geolocation, improved calibration. We should have the full record available by the end of the year. Doing your pointing from the International Space Station with this new pointing mirror system turned out to be a little more complicated than we maybe anticipated. We had to rederive corrections for the mechanical system, which are hard to characterize on the ground. And we also needed to have some additional SRU data to account for that flex of the GEM EF relative to the center of the ISS. And our SRU didn't work during the whole mission, so we ended up borrowing some data from Cowlette, which turned out to be a great way to get the improvements we needed. This figure shows you coastlines and some early data where the dark is water and light is typically land, and you see there's mislocation. When the updates were included, we have much better agreement. And if you try to put some numbers to that, you can see that the early data had errors upwards of 2 kilometers in these SAMs, and now that's generally under 300 meters. We validate with the TCON data. The colored dots are showing the OCO3 data compared to TCON, and then the grayed out dots are OCO2 compared to TCON. So we've got this nice common standard that both of the mission use to help us have consistency between these data sets. So in conclusion, we're 2 years into our 3-year mission. We've learned to operate in the dynamic environment of the ISS, have a new data set coming out now with significant improvements on V-early, and enjoying the co-collected data from many of the payloads like EcoStress, JEDI, and PRISM that make it great to be on the ISS. Thank you.
Greetings from the SAGE 3 ISS team. My name is David Flitner, and I will be presenting Chemistry and Climate Changes in the Earth's Upper Atmosphere as seen by the Stratospheric Aerosol and Gas Experiment 3 on the ISS. You can see the payload in the center of this image, which is located on the starboard side of the ISS. Stratospheric Aerosol and Gas Experiment, or SAGE for short, has science objectives to monitor the vertical distribution of aerosol, ozone, water vapor, and other trace gases in the Earth's stratosphere and troposphere to enhance understanding of ozone recovery and climate change processes in the upper atmosphere. Utilizing a mission approach of a hosted external payload on the ISS with the methodology of solar and lunar occultation for monthly coverage of the tropics and mid-latitudes since June of 2017 to produce vertically resolved profiles of trace gas concentrations and aerosol extinction values on a monthly basis. The mission team is led by NASA Langley in collaboration with NASA Johnson, the ISS program, and the European Space Agency and Thales Space Italia. An example of the ozone data is shown here for the tropics, where the variations are not so disturbed by changes in weather patterns in the upper atmosphere. Still, nonetheless, there's year-to-year variability. It's influenced by slight changes in the circulation pattern. So if we remove the annual cycle and get a picture like on the right-hand side, and you can see there's anomalies that propagate from above and downward with time. And these anomalies are in phase with changes in the stratospheric wind patterns. These changes in the stratospheric wind patterns are the quasi-biennial oscillation, which is one of the dominant forces of natural variability in the stratosphere. Changing our focus to water vapor, it plays many roles in the Earth's atmosphere, but an important one is that it is actually a greenhouse gas affecting climate, and it's also key to many chemical reactions. A major pathway for water entering into the stratosphere from the troposphere is through equatorial convection. And on the left-hand side is a time series of SAGE observations in the tropics. And you can see there's alternating wet and dry periods. And you can see these periods, this dry air, enter the stratosphere and then propagate with time upwards. And this is just the regular seasonal cycle that displays itself in the tropics. Again, if we remove that seasonal cycle and then look at longer term variations, we see periods of moist and then dry on longer scales on the order, again, corresponding to the QBO phase, which is roughly about 28 months, but it has a pattern that's different than ozone, because here it's how the QBO is affecting the temperature as the water enters from the moist troposphere into the dry stratosphere. Since Stage 3 ISS has been up, we've seen several changes in the aerosol distribution. Here we've got data illustrating that for time and location. At the beginning of the record, there were wildfires in the Pacific Northwest that injected a record-setting amount of smoke into the stratosphere, followed by that of an eruption of a volcano in the tropics, and then one at mid-latitude in the summer of 2019, coupled with some fires that were raging in Siberia, enhanced significantly the stratospheric aerosol loading. And then that was in some sense eclipsed by the stratospherical injection of smoke from the Australian wildfires that became the new record-setting benchmark for wildfire injection. Thank you.
Video from the International Space Station by Brian Walls, Deputy Program Manager of the NASA Imagery Experts Program based at the Marshall Space Flight Center in Huntsville, Alabama, and Carlos Fontenot, ISS Imagery Manager, and Chris Gateau, ISS Imagery Specialist from the Johnson Space Center in Houston, Texas. The video capabilities of International Space Station have continued to develop since launch, with a maturing core capability ready to support both the operation of the flying laboratory and the scientific and commercial payload users of the facility. Beyond that core capability, there are opportunities for more unique and developmental uses of video imagery. The full presentation looks at the existing and near-term roadmap of upcoming capabilities and an overview of some of the innovative uses of imagery that have been recently demonstrated, including ultra-high definition or UHD video, 360-degree video capture, and augmented reality applications. The video capabilities of International Space Station have continued to develop since launch, with a maturing core capability ready to support both the operation of the flying laboratory and the scientific and commercial payload users of the facility. Beyond that core capability, there are opportunities for more unique and developmental uses of video imagery. The full presentation looks at the existing and near-term roadmap of upcoming capabilities and an overview of some of the innovative uses of imagery that have been recently demonstrated, including ultra-high definition or UHD video, 360-degree video capture, and augmented reality applications. A common source for the core downlink channels is the handheld video camcorders. For several years, the Canon XF305 camcorder has been the primary multi-program device available for astronauts to capture video in SD or HD formats live or recorded on the device. In 2021, transition to the Canon XF705 will support ultra-high definition, UHD sometimes called 4K, and HD formats. Initially, support for UHD will only be for a single live channel for pre-planned outreach content, but recording in UHD on the camera will be available as needed. Outside the station, there are both HD and SD cameras installed, some with pan-tilt zoom capability. U.S. spacesuits have 3 video cameras that can operate during extravehicular activity, or EVA. Originally all SD, 1 camera on each suit is being replaced with an HD unit. The large external robotic arm, Canadarm2, has 4 associated cameras. All of these sources share the 6, soon to be 8, core download channels along with other sources, such as visiting vehicles like the SpaceX Crew Dragon when it's docked. Despite the large number of potential sources, though, oversubscription of the downlinks is rare except for during docking, spacewalks, or robotic arm operations. U.S. spacesuits have 3 video cameras that can operate during extravehicular activity, or EVA. Originally all SD, 1 camera on each suit is being replaced with an HD unit. The large external robotic arm, Canadarm2, has 4 associated cameras. All of these sources share the 6, soon to be 8, core download channels along with other sources, such as visiting vehicles like the SpaceX Crew Dragon when it's docked. Despite the large number of potential sources, though, oversubscription of the downlinks is rare except for during docking, spacewalks, or robotic arm operations. Besides the core capabilities, individual experiments or other payloads can fly their own imagery resources and download video, potentially using one of the core video downlink channels, or by using Internet Protocol resources to stream video or download files. There's even the option of recording the video to media and returning the media to the ground, though cargo return capacity is even more precious than delivery to ISS. Choices made in the design phase can have an impact on the cost and function of a payload. Being aware of the available resources and the constraints and requirements involved can ease implementation. Here are a few examples of payloads that supply their own imagery. Both content and destination of any video are important to the choices made in payload design. For a camera sealed in a proprietary experiment rack, the video could go directly to the owner with no need for anyone else to view it or have a copy, while video that may have a view of astronauts or someone else's intellectual property, or that has been paid for by the government, will need to be reviewed before any public release and may need to be archived. If live video is embedded in a data stream going out to a remote user, the design must accommodate getting that video back to NASA Johnson Space Center for review in real time. Using one of the core downlink channels avoids that requirement. The ISS is a unique environment for research with a rich video system to enable the mission and give eyes to us below. If you want to know more, watch the full presentation or search for NASA ISS Research in your web browser.
All right, we're back here live with Kenton and all the panel. So panel, if you'll please turn on your cameras, keep your microphones on mute until you're answering Ken's questions. And I look forward to hearing the discussion. Back to the audience, please go ahead and type in your Q&As. We're getting a good number of Q&As. So hop in there and let's keep up and fill up the hour. Thank you.
Thanks, Alan, and thanks to the 3 presenters. Those were great talks. And if you aren't aware, you can see the full longer talks on the ISSR&D website also that has the more in-depth presentations. So today we've got David Flitner, Anne-Marie Eldering, and Brian Walls from the 3 presentations we had today. And so what I'm going to do is walk through the Q&A that we've gotten so far, and then folks keep sending them in as you have questions. And if not, I have a few of my own also to ask to get the discussion flowing. So thanks for the presentations, and I'll start out. The first question is for you, Brian. Liam wants to know do we anticipate any changes to the public streaming side of video from the ISS, specifically EHDC 6, which is the HDEV channel. It appears it's streamed in 480p, and they're curious if there's going to be any updates to that.
So we've had quite a bit of discussion about that. We would really like to be able to take it to an HD channel, but the current issue is having a 24/7 available HD content. The external HD cameras on the station, except for the one that's used as a backfill for the HDEV experiment, are normally not left on all the time. And there's concerns about the fact that they're doing that it's heat damage and just the constant use may reduce the lifetime on the cameras. So during the evenings, during crew sleep time, they don't want to use any internal camera because of privacy issues. And what they normally do is switch to the external cameras. And Liam, I see you mentioned in the chat that EHD6 is there, but because it's coming down for HDEV, it's not available to also come down through the other channels that we're using. So unless we were just to mirror the content, and then somebody would have to watch it. So that's a long answer, but the shorter answer is we would like to do that, but we need another source to have a 24/7 HD content that we can stream out publicly.
And Brian, I have a follow-on question for that actually. With the new commercial module that's being planned from Axiom, how are these data channels being planned for accommodating cameras on these commercial modules or this upcoming commercial module? Has that been discussed at all?
I have not heard a discussion of that. So there is precedent that visiting vehicles that have video on board, they can connect to the internet on station and are able to download video using the SpaceX Crew Dragon, for instance, can have video from their system coming down on one of the 6, soon to be 8 channels. I would expect that they would be able to do that, but they would also be free to have their own downlink channels. And I'm not sure what their plans are.
Thanks for the answer. So folks keep upvoting questions or liking questions in the Q&A. I'm just going to go in the order that we've received them. And so the next one, I believe, is for Anne-Marie. The ABET has a question that appears to be asking if there is a link in emissions and the wildfire seasons that we've been seeing, the really intense wildfire seasons the last couple of years. And I'm curious if you or anyone from the OCO3 team has looked at those links.
So thanks for the question. It's a little bit out of my field of expertise. We really focus on the carbon dioxide and understanding the carbon dioxide in its natural cycle, but we can certainly say that the warmer and drier weather we see in response to phenomena that's both driven by El Nino and also driven by climate change is definitely an ingredient to the higher frequency of wildfires people have seen in recent years. So there's a connection of carbon dioxide to warmth and dry weather and changing climate, which links to fire. We haven't done, we have some people in science teams who've done studies mostly from the OCO2 data. For example, when there were big fires over Indonesia, they could actually learn about how much the carbon release changed with the fires that basically burned down the trees and released the carbon dioxide. So you can see the CO2 showing up in the atmosphere when these very massive fires happen. Thanks.
Thank you for the answer, Anne-Marie. Next question is back to Brian. And it's curious, it looks like Anna has a question about the South Atlantic magnetic anomaly and how that may have effects on the external cameras. And I'm not sure if you're familiar with that, but.
Yeah, I am not particularly familiar, and saw she had asked if there had been any studies, and I am just not familiar with the background on the studies. As far as the radiation environment and the effect it has on the cameras, it is pretty significant. We generally have to change out the cameras on station after about a year of use. And it varies by camera and from time to time on just how quickly you'll start seeing bad pixels, which typically shows up as colorful pixels. Looks almost like a star field after a camera has been used for a while. And so we try to swap them out. And then one of the interesting things that was found with the HDEV experiment, which was a camera on the, it was a set of cameras that weren't particularly fancy cameras, and they were on the outside of the station, and the expectation was that they would have problems very quickly, and they never did. And we're still sort of scratching our heads on why that was. And it ended up failing for a completely different reason, not due to damage on the camera sensors.
Thank you for the answer. I have a question actually for David and the SAGE 3 team. I'm going to jump in with one of my own. Has the SAGE 3 team looked at, so in your presentation, you discussed the 40 years of SAGE 2 data looking at aerosols. And I'm curious, we do a lot of imaging for wildfires through the ISS for disaster response. And have you guys looked at any trends? You said that the recent Australian wildfires were the most intense on record. But I'm curious, are there any trends that you've looked at in aerosol emissions as it links to wildfire seasons in the SAGE 2 data and then the recent SAGE 3 data?
Yes, so yeah, that is a very timely question. The team has actually in the past gone back and looked at this SAGE 2 record for occurrences of these intense injections of smoke, if you will, into the stratosphere. And there are a few events. Certainly the wildfires in the Pacific Northwest were much larger. They eclipsed anything from the SAGE 2 time period. And then so when the Australian wildfires happened, it was even much greater. One thing to note is those fires in Australia have been burning for months and months, but there just hadn't been the mechanism to inject the smoke into the stratosphere to basically get up this kind of thermal barrier up into the stratosphere, where then it can just stay there for many months in time, because there isn't really the precipitation process in the stratosphere to cleanse the atmosphere like we have in the troposphere. So this is a topic that we're looking at. I will say that during the past SAGE 2 record, which ran for 20 years, the biggest aerosol event in that was a volcanic eruption at Mount Pinatubo in the early 1990s. And in some sense, that kind of masked the ability to see some of these smaller events. The wildfire events that we've seen are comparable. These record-setting ones are comparable to what I'll call a minor volcanic eruption. So on the big scale of things, the volcanic eruptions are still the items that really perturb long-lasting climate sense, the stratospheric aerosol layer. But we do know that just from studies from that, for various minor eruptions, if they occur frequent enough, then even that small amount needs to be taken into account when you look at the energy budget. For example, in the early part of this decade, realized that there was this gradual kind of trendward upward in aerosol in the stratosphere, and you needed to actually take that into account because it accounted for about a third of the warming. It would offset about a third of the warming you would have expected to CO2 increase over the same decade time period. So as we want to understand how policy decisions have a result and the efficacy of we need to understand some of these other details. And so the data that SAGE provides helps atmospheric scientists and modelers as well. Thank you.
Thank you for the answer. I have another question for Brian here, which is whether you guys use any 3D cameras on the ISS.
So we did fly a 3D camera and did some footage. As the 3D craze and all of the 3D televisions and all of that kind of dropped from the public view, there was really no longer interest in shooting it. But we do have some footage, and if you look online, look on YouTube, you can find some of that footage if you have a way to play 3D content. Similar as there's some 360-degree content that's a couple of different folks have flown 3D cameras, one a fairly similar, a couple I think that are fairly simple all-in-one solutions, and then a larger time teamed with a Felix and Paul Studios and flew a cinema quality 360-degree rig and has been putting out some really amazing footage from that.
Thank you. And a follow-up question from Jennifer asking, you mentioned that the cameras are swapped out after a certain period of time. Are they brought back down to be studied by the team to see how they were affected by their time on the ISS?
Some of them have been, and for the most part, at this point, when you're talking about just the internal cameras, either the still cameras or the camcorders, they aren't brought back down because it's more expensive to bring things back down than it is to fly them up, and that's expensive on its own. So unless there's something specific that the vendor wants to see, typically they aren't. But because of that thing, we've worked with the folks that build the cameras to get them back units to see what the damage is.
Thank you. We've got another question, I believe for Anne-Marie, and possibly also David. The asker is wondering what sort of sensors can be used to detect black carbon?
I don't know if Dave wants to take that one. Certainly, so from just to help people understand how we make the carbon dioxide measurement, we're looking at light that gets absorbed by the gas and changes the light we measure in a very specific way. So you have absorption lines that are very specific to gas molecules. The black carbon is actually a small particle, an aerosol, and it's black because the way it interacts with light, which is actually hard to characterize. So maybe Dave has a few things to say, but certainly the measurements we make are really about gases, not about aerosols. But Dave's about aerosols, so I'll let him take over.
Thanks, Anne-Marie. Yeah, well, the principles are much the same, but you're right, the spectroscopy is different. So the materials, how they interact with light is different. Aerosols can be more difficult, or solid matter is going to a little more broadly based interaction, not so discrete as you might have with gas molecules. For black carbon, I don't know if there are any prominent absorption features. Certainly there is some broad spectral differences between visible and near infrared and then infrared. So using that broad spectrum, putting different parts of the spectrum together can help detect some of the difference. And also how then maybe there is some scattering, and then how that embedded within the air, and how scattering goes on in the air, and any particle introduced in there to absorb light like black carbon would do. And so that then how that absorption or that reduction in light decreases the amount of scattered light, and then that has kind of just a standard wavelength dependence as we do with what we call Rayleigh scattering or molecular scattering. So we kind of have to really use our modeling techniques and use broad pieces of the spectrum to understand, to discern black carbon from other kinds of particulates.
Thank you, David and Anne-Marie. And I actually have a follow-on question for you, David. In your slides, you were showing the aerosol extinction, and you compared it was a pre-calibrated, a estimate pre-calibration from the models and then post-calibration with the SAGE data used to help calibrate it. And I'm curious, it mentioned a bunch of other sensors that were measuring aerosols, but they obviously have a difference in the measurements that you guys are providing from SAGE. What makes your guys' measurements better to use for these sort of calibrations?
We like to say it's a direct measurement of the aerosol that's in the atmosphere, how much it attenuates. Our fundamental measurement is how much is light attenuated, the light from the sun, how much is attenuated by the atmosphere. We have to account for attenuation by these gases, as Anne-Marie mentioned, as well as just regular air molecules. But what's left over, we can subscribe to aerosols. And so we have a direct measurement right there. The other measurements that were mentioned rely upon scattering. And so they enhance your signal. For example, the active one, Calypso, is a LiDAR. It sends out a pulse of light, and then it receives back light that's been scattered. So it's get an enhancement of light, but it doesn't know, it gets a little more complicated because how much light it gets back depends upon how much there is, and then what the shape of the particles are and what they're made of. Whereas our measurement of extinction really comes down to how much stuff there is. There is some dependence upon the kind of stuff, whether it's smoke or whether it's sulfate particles or how big they are, but it's primarily how much stuff there is. And so that provides us a measurement of extinction. And that's a fundamental parameter needed by the climate models that then try to model what's the effect of a volcanic eruption, or what's the effect of smoke injected in the stratosphere. So that's kind of a more fundamental measurement. You can't really come up with one. And I will say it's been kind of difficult then to make, how do we compare our results? How do we validate our results versus somebody else's? Well, there hasn't been really a standard to do. So then we have to do kind of internal validation. But nonetheless, we're still kind of trying to be good scientists and skeptical of our own results so that others can use them. I hope I answered your question more directly than it seemed like. Okay.
Thank you for the great answer. So I have another question for Brian, and then I'll add a little bit from my own experience after your answer, Brian. Elizabeth wants to know if we have an idea of how the crew feel in terms of more of an emotional, personal level when imaging Earth or taking video of Earth. It's kind of a tricky question to answer, but see if you have an answer, and I have one I can help with after.
Well, I've certainly heard a lot of them talk about the feelings that it engenders, and for many of them, their favorite, thing to do in their spare time is to go to the cupola or a window and look down at Earth, and they call it the overview effect. Saw there was a question. The question was specifically about putting sensors on them and measuring, and that could potentially be an experiment if someone wanted to pitch it and create it. Clearly, it would need the participation and interest of the astronauts to be willing to let people know what they're feeling. And perhaps the easiest way is to ask them, because many of them are happy to talk about the feelings that looking at Earth creates.
And I'll add, we do all of the crew trainings for the handheld imagery and the debriefs with them. And actually, I agree with you, Brian. You could probably figure out a sensor and do a payload for it, but honestly, the best is during the debriefs when we ask them and they give us how they felt and what they thought about doing it. And as you can expect with any person, it varies heavily person to person, but the general consensus for most crews, they really enjoy doing the imagery of Earth, and it's one of the things that they like to do the most. So we have another question, and I believe this is for David or possibly Anne-Marie. It says, did NASA test or have you any experience using the HAWC/A or OAWL sensors for aerosol detection? I'm going to go to David first.
I guess I will say that I'm not familiar with that acronym. So no, I'm not familiar, but that doesn't mean that there aren't other parts of NASA. I'll have to look into that.
It's a type of high spectral resolution for LiDAR for aerosols, and I believe it's based on the heritage that Calypso used, or similar to an instrument that Calypso used.
I mean, there actually is an endeavor, and so I don't know whether they're using that technology, but there is the next generation of aerosol measurements from space as part of the, I'm not going to get the acronym right, but anyways, we've got a next generation, say Calypso, again, to understand aerosols in the context of also clouds and convection, understanding the precipitation process and climate.
Thank you. Anne-Marie, I have a question for you actually. So my own work, we are often sending targets to the crew, right, to image a certain target on the surface, and we often have to battle with how far off nadir can a target be when we still collect good data. And I'm curious, you talked a lot about your SAM modes and your imaging. What difficulties do you have with operating on the ISS versus the previous instrument, which I believe was satellite-based? And how does that help or affect your targeting?
Yeah, thanks for the question, Kenton. So the measurement mode I talked about, the SAMs, was made possible by this pointing system that we have to be on the ISS, because the free flying satellite moves the whole payload when it wants to look at different locations. So it can look anywhere at once if it can point the satellite. On OCO3, we're there on the GEM-EF and we use a pointing system. And the biggest thing we have to be worried about is that if light bounces off of a bright object and into our instrument, that could potentially damage things. So we have to be aware of where are the solar panels, where are the other payloads, where's the robo arm, etc. So we actually have a keep out zone, an area we don't look into because of potential obstruction. So that's limiting how far off nadir we look with the pointing system, and therefore defines the area we can use for the SAMs. So that has limited the number we can collect, although we also worry if you look really far off nadir, you look through very long paths of light and footprints get very skewed. So you probably wouldn't want to look too much further than we do, but it's constrained, and the solar panels are actually the biggest thing we worry about, because if you've ever watched the time series or movies of the ISS, you know how those solar panels rotate through many axes, and they're very dynamic in their movement. So we don't go anywhere near where they're going to be.
Thank you. Yeah, it can be difficult working on the ISS, and this is kind of something I want to ask David too. Is Sage 3 is also a follow-on to a satellite mission, and so similar to what Anne-Marie was talking about with the trickiness of working on a space station as an external payload, where you've got a lot of other things going on. What difficulties or what workarounds have you guys kind of dealt with to work in this environment versus a satellite?
So definitely it keeps the operations team busy. Just as Anne-Marie mentioned, we have to be constantly looking at what the IS is going to do at all, I think, is basically the timeline of what kind of operations, when will things be docking, what orientation will we be in. Until I was part of this mission, I never realized we fly upside down, fly backwards, and there's a whole range of orientations that you usually don't get into with a free flyer satellite. So that really certainly keeps the operations teams has to keep in touch with the ISS program as to what's going on. For many of we're able to take observations, kind of plan them, and if something gets in the way, then it's a matter of us to do quality control. We don't have quite the sensitivity that Anne-Marie was mentioning. Like many of our occultations of the moon get interrupted by solar panels because it's on the night side and we're looking off to the side or something like that. Or maybe the robo arm came by and we weren't quite ready for it. So that's the downside. So a key upside is this, that it's on a manned spacecraft. For example, we had our main power interrupted twice, once for a breaker basically went out, and astronauts were able to fix that. Whereas the first stage instrument was launched on a satellite when the batteries failed after about 2 years or something like that. And so that was the end of the mission. The Sage 3 Meteor was on a precursor, a Russian orbiting satellite there. There was a failure related to the power system. So again, something broke, there's nobody to fix it, there's no way to fix it. That was the end of the mission. The instrument could live much longer than that. It was the spacecraft is usually the limiting issue. But with ISS, in a sense, the crew was there to do experiments, but also maintain. So I'd say that's an upside.
Thank you. Definitely something that is not often considered is it's also, you know, cheaper to fly a payload like this to the ISS, right? A full satellite mission is quite expensive. And I can't remember if it was Stage 3 or OCO3 that discussed using a spare imager set from a previous mission to fly. And that's something that's capable when you have all these other components are provided by the ISS. You get power, you get all this stuff. You can take these spare parts and get good data from it from orbit. So we have another question, it looks like, for Brian. Could you speak more about augmented reality, the augmented reality device on the ISS that was mentioned in the presentation? I believe you guys were talking about the generation of augmented reality.
Yeah, I didn't really cover that much in the presentation either. I'd intended to. I know that they are using a headset for some crew training, specifically allows the use of some whiteboarding, and so it can be very useful for like walking. Somebody on the ground that's walking the astronaut through a procedure can see from their eyes what they're doing, and can even do things like draw circles. And some of that is currently rolling out. I know they're doing some testing on it currently.
Thank you. Looks like we have a question for David, I believe. User asked, this goes back to our black carbon discussion, if you can go for the UV spectrum when doing black carbon detection.
Well, it depends upon where in the ultraviolet, because there are other things going, other constituents in the atmosphere that absorb light. You get too far. I mean, you go into the UV, A, B, C, you've got ozone absorption that mask things. But there is an aspect of ultraviolet light, UVA, the scattering in that region, scattering by molecules in the atmosphere. So there's a lot of scattering that goes on. And if you introduce particulates in there, such as black carbon, that absorb light, basically take light out of the scattering process, then you're changing the wavelength character compared to what you would have for a purely molecular atmosphere. And today, that's actually a physical effect that is utilized for a series of sensors that were designed just for ozone sensing. Tom's series, Total Ozone Mapping Spectrometer, and there were follow-on instruments to that, were designed just for ozone sensing, but because of this way it changes the wavelength behavior of the scattered light when you have particulates in there, such as black carbon. So in a sense, yes, directly, it can be, to use the UV spectrum directly is not so easy because it's got, in a sense, interfering gases and species.
Thank you. I have a question of my own for Anne-Marie. So during your presentation on one of your charts, it showed the different classes that you were measuring emissions, and one of them was for eco-stress targets, it suggested, or looked like. And I was curious, I'm familiar with eco-stress payloads, another one of the Earth observing payloads on the ISS. What's that, how are you guys working together? What's that collaboration?
Yeah, so the eco-stress team, when we used to go to the office, if you all remember those days, the eco-stress team was just across the hallway. So we have physically very easy communications with that group, and there's some scientists that really see the value of these sets of data being used together, because eco-stress is interested in what we call evapotranspiration, or how plants are taking up water and releasing it to the atmosphere. And we're interested in the carbon dioxide, right? So the plants take carbon dioxide out of the atmosphere when they're growing. And I didn't mention this in my presentation, but there's actually a signal we detect called solar-induced fluorescence. So when plants are doing photosynthesis, they release a little bit of light, and we can actually find that light in our measurements. So we can see a sign related to photosynthesis, the carbon dioxide that they take out of the atmosphere, and then EcoStress sees the water transport through the plants. So when you get those three ingredients together, you can really start to learn more of what happens to a plant when it's hot and dry. What's a plant do when it's raining just the right amount? Does it change behavior maybe when it's really wet? So the scientists are using those data together to look at plant performance. So it started out as just kind of a, recognizing EcoStress was going to be on the space station, thinking about where they were targeting to get their data. And so we added some of their targets onto our target list so we can co-collect the data. So we're just starting to see some of the results roll out from that, but it was a pretty natural collaboration. And then of course, JEDI's up there too. We don't happen to have JEDI on our panel today, but they're looking at how much plant material is there, right? With the measurement of the total amount of biomass. So you can imagine scientists in this field just see this as a great opportunity with ISS measurements.
And that's a good point, JEDI. For the folks listening in here, these presentations alone aren't the only Earth observing payloads on the ISS, and there's a lot of information out there if you go search on the internet. There's a wide number of payloads that are doing Earth observations that have a lot of overlapping interests and research areas that really make and highlight the benefit of having a space station, right, that you can have as a national lab that does this sort of research. I had another question here from a user that says, I believe this is for Anne-Marie. Do extra emissions cause degradation of the signals?
Yeah, so in terms of our carbon dioxide, the extra emissions of the gases, of the carbon dioxide gases, don't really change our ability to make the measurements. They certainly contribute to what we see when we have a multi-year record, and that's that carbon dioxide concentrations go up each year. So we can see this extra 2 to 3 parts per million growth in the atmospheric concentrations due to the extra emissions. But in terms of the technique or our approach to measure, extra gases don't get in the way. Those aerosols that Dave loves so much do cause us a problem sometimes. When it's very cloudy or very heavily polluted with aerosols, that light scattering Dave mentioned is difficult for us to account for in our retrieval. So when we see very heavily polluted or cloudy regions, that's places where we cannot make measurements. So the clouds get in the way for us.
Thank you. And I'll pose the same question to David then. Do emissions affect your ability to do your measurements with SAGE 3? David, I think you're muted.
Yep, sorry, I've got the double process here. Yeah, so quickly, so emissions into the stratosphere, most of what we talk about for emissions is at the tropospheric level, and many things just don't make it up into the stratosphere for one way or another. Part of that's because of the precipitation process tends to clean out the atmosphere. Gases like chlorofluorocarbons, which were essentially inert in the troposphere, could make their way up into the stratosphere after being released after years, and then ultraviolet light would break them apart and release the chlorine radicals, and that then would produce the signals that we saw that decrease with time of ozone. So no, it's actually, we're kind of looking for that sort of thing. Anthropogenic or human caused, it's not so easy to see in the stratosphere. That's always been a question. Is there industry in, say, Southeast Asia, are chemicals from that region making its way into the stratosphere and altering the type of stratospheric aerosols we have? That's kind of a question we'd like to answer. We just don't have enough data on that, or the right kind of data. So for the stratosphere, it's not so much, we don't see that as often. So I'll leave it at that.
Thank you. I think we have one last question here for Brian. It appears, George, has a question about comparisons between SOI and CMOS sensors as it relates to rad hits. And have you guys looked at comparisons between those two types of sensors? And what goes into your analysis process?
Yeah, so we've looked at several different sensor types and had opinions on, hey, this one ought to be more resistant than that one, and reality hasn't correlated very well. We do tend to see that, if you fly a camera and you see if it gets a lot of damage or it doesn't get a lot of damage, and then you fly another of the same model, it tends to look similar. So it must have something to do with the way they're being built, but then sometimes you'll get one CMOS camera that does great and a different model does really poorly. So there's still some room for research and figuring it out.
Thank you. So we're coming close to the end of the session here, and we've run out of our Q&A questions. So I have one last question I want to pose to each of you. We'll start with Anne-Marie. So what do the next couple of years look like for the OCO3 payload? How long is your guys' mission planned for, and kind of what's that outlook?
Yeah, so we had an agreement for a 3-year mission, and our official start was August 2019. So August 2022 is nominally the end of the mission, but it looks like the next payload that's going to come use the GEM-EF spot we're in is a bit delayed. So right now we're hearing we might be able to stay till January of 2023. So we have a little bit extra time on the mission. We're really honing, as Dave mentioned, the mission operations teams keep busy. So now that we've got the wrinkles ironed out, the plan is to just do the smoothest operations we can. And then also we typically want to get the best calibration and the best processing so we can deliver a final data record at the end of the mission. So the other thing is just doing a lot of science with the data and engaging as many people we can and making good use of the data. So the plan for the remaining time in the mission is collect as many good observations as we can, get the data in people's hands so they do science with it, and then provide the best end data record. It'll probably take us about a year after we're removed from the ISS to wrap up everything and get that last loaded data delivered. But it's a pretty different experience when you work on free flyers that work as long as they work. Sometimes missions end up running 5, 10, 15, 20 years. So it's really odd for my team when I say, okay, guys, like this is what we're going to do in 2023, and then you're going to be done. And they're kind of surprised, but we're excited. We're having a lot of fun being there. We look forward to the year that remains.
Thank you. Glad to hear it. I'm going to pose the same question to you, David. I know Stage 3 is on its extended mission, and I'm curious, you've got the next couple of years for that, and what are the plans? Is there another extension possible, or what's going on there?
Yeah, the SAGE data set really, it has its most value the longer it is. It's climate, so looking for small changes over long periods of time, as Anne-Marie's team is doing. We want to continue to make as many measurements as we can to build the quantity of data for the science community to use. We've just been renewed, as you said, for another 3 years. We were told to plan for 6. NASA Science has a policy where they have the cycle of every 3 years. They review their operating missions, and each mission that wants to continue submits a proposal. Within that, I would hope that as long as ISS is willing to have us, we're on the manifest, that we'll make the science case with NASA headquarters and hopefully get renewed. Again, fortunately, we've had this, we have a spot that's maybe not so prized as a spot there on GEM, but go as long as we can, and hopefully all the other instruments have outlived the satellite. Well, I guess we'll see if we can do that here as well.
And that's a good point that both David and Anne-Marie have had made here about getting as much data. People don't often think about it when you have a payload on the space station that's collecting data for a finite period of time. Your main objectives are typically, is it getting the right good data? And then, okay, how much can I get? And the actual research ends up falling to the after you get the payload back, because you just want to get as much as you can while you're up there. And then you start digging into it, making sure you have the good data beforehand, but then you start digging into it. So it's really just as quick as you can go, as fast as much as you can get before you're either replaced, or if you do get extended, that's great. We are up with about 1 minute left here, so I think unless Alan, if I have the time wrong, I think we are at the end of the session. I wanted to thank Anne-Marie, David, and Brian for great presentations, and also the audience for a lot of great questions and interaction. I thought this was a great session, and I appreciate everyone participating this afternoon.
Well, if you want to get Brian's answer on your last question, we can do that.
Oh, I don't see it in the Q&A anymore.
The question you just asked yourself.
Brian, if you saw it.
The answer that you asked the other two panelists.
Oh, sorry. I passed over you, Brian, because I already know that you're not a payload that has a short life, and you're going to be running the cameras on ISS for a long time. But what are the next few years of plans for the cameras?
Well, in the short term is we're going to be moving to cameras inside that are capable of UHD, so some higher resolutions. Certainly possibilities for doing other things as well. I expect to see that the imagery will continue to grow as the technology does. Also, of course, ISS is a great platform for testing equipment that we're planning to take to the moon and then to Mars. So be expecting to see definitely working now on choosing what's going to be the handheld camera that they'll be using out on the moon and on the way, and probably be flying that to ISS to get some time in space with it.
Thank you for the answer.
All right, Kenton, thank you very much. And Brian, Anne-Marie, and David, thank you. This has been great. It's the nicety of having a small panel is that you can go through a lot of material where you can't if you have a panel of 5 or 6 people. So this has been very nice and very wonderful. And I know that our audience has appreciated it. So I got some closeout work to do. First, I need to thank our sponsors. And that is the Boeing Company, this year's ISRDC Platinum sponsor. They have been that for the 9 previous ISS Research and Development Conferences. And of course, Airbus, KBR, Leidos, Northrop Grumman, Sierra Space, Space Tango, Teledyne Brown Engineering, the Engineering and Innovation Technology Development Team at the University of Alabama. That's a mouthful. And thanks to the exhibitors, Jacobs Oceaneering and Tech Shop. Please come back tomorrow for our second day of these excellent tech sessions. We start off at 0700 GMT. That's Zulu time, or 3 a.m. Eastern, a session on commercialization. Most of the folks on commercialization are in Japan. So that's the reason we're doing it at that time in the morning. So if you can get yourself up, please chime in and listen to what they have to tell us. Then later in the morning at 9 a.m. Eastern, we'll start with sessions on human health in space and several others. So please join us. And with that, I thank you very much. Hope you have a good, safe evening and good night.